Day 2 of our HABs Delta workshop. I'm going to just kick us off with a quick recap of yesterday and then move us into our presentations for the day. We have a packed day today as well. Next slide, thank you. Next slide after that, actually. Okay, so really quickly, just some high-level take-homes from yesterday. I feel that we had a lot of take-homes, but these are just a handful that I captured to get us started today. Yesterday, we discussed kind of our top interests for coordinated monitoring. Our Menti poll in the morning had the top answer with better understanding drivers, perhaps in the Delta. We discussed a little bit the surprising answer around having an increased public awareness of harmful algal blooms ranked the lowest, although people didn't really get a chance to rank all of their hopes for coordinated monitoring in the Delta. So this is an area to resolve and kind of think about more as we develop this strategy. Additional themes that were repeated throughout the day, decision-making on different timescales, how that influences data and communication needs. That's something that we're going to think about today in the breakout rooms as well. Where do I fish today versus like, how do we understand drivers over time? Again, funding for all of the things surrounding HABs monitoring in the Delta, that was brought up repeatedly. An additional point that we will continue to think about, how we will standardize the approach to monitoring and how that helps all stakeholders in their decision making. And we also acknowledge the fact that we can't really do this in a single effort. We are doing this 2-day workshop, but really we're going to have to continue to come back to this effort and continue the dialogue on this into the future. So next slide, please. So quickly to put our management questions up once more. Our primary management question driving the monitoring strategy is how can HAB monitoring efforts be collaboratively designed, facilitated, integrated, performed, and or standardized to achieve status and trend monitoring objectives and to fit the scale of management actions, timing of ecosystem processes, and climate change challenges. We also have another question, which was what are the drivers of HABs in distinct areas of the Delta to inform management actions? Next slide. So yesterday we talked on the collaboration piece, and next slide, please. Next slide. And today we're going to dig a little bit more into the data sharing and integration piece. So a couple questions that we're thinking about today are how can relevant harmful algal bloom data be made accessible and available? And how can current barriers to data storage and access be overcome to support a comprehensive monitoring program? And with that, I think I'll hand it off to this morning's moderator. Thanks, everyone.
Good morning. My name is Jenna Rinde. I'll be helping moderate this session. Our first presenter is going to be from Bouma-Gregson, and he'll be informing us about information of how to monitor in the system and tools, and looking forward to his talk. It comes. Sorry.
All right, hi everybody. Oh, great. All right, so great. So my name is Keith Bouma-Gregson. I'm at the U.S. Geological Survey, and I'm going to be kind of giving a bit of a rapid fire overview of different types of monitoring tools and technologies for collecting data relevant to HABs. And this is not necessarily an exhaustive list. I packed this pretty full, so I'll try to be moving quickly, but I know the slides will be available, so hopefully this is also a good resource for folks. Okay, you can go to the next slide. Great, so I'm a part of the U.S. Geological Survey. We are a non-regulatory environmental science federal agency. Our goal and mission is to provide data and scientific analysis to managers to help address the water issues, challenges facing California. And we do this by working really closely with state, local, and other federal agencies coming up with projects to make sure that work we're doing is relevant and gives them the information and analyses they need to answer the questions and make the decisions that they need to make. Slide. And I also want to do a quick shout out and thanks to Christine Jove at the IEP. Some of the slides and content I'm sharing with you today are based off of a deck that she put together for a Central Valley Regional Board HABs workshop a couple of years ago. So thanks. I'll share the content. Fine. All right. So yesterday we talked a fair bit about how we measure is really dictated by our monitoring goals and what we're the reason that we need to collect data. And so I made this sort of like set of concentric circles about the parameters that you might monitor. So at the center is designed to be usually what we're most interested in. So that would be like often it's toxins or taste and odor compounds if you're a drinking water provider. But sometimes you don't have the funding to actually make that measurement, or sometimes the sensor just doesn't exist for making the measurement how you want to measure it. So then there's different levels of other measurements you can make that might not be exactly what you want, but might still be providing relevant data. So stepping out, you might then make a measurement about cyanobacteria, which are often the organisms that are producing these compounds. Stepping out an even further away, you might be making measurements just on the overall algal or phytoplankton community or abundance, where you don't necessarily know if it's cyanobacteria or toxic, but at least you're getting some estimate of how much biomass is out there. And then moving out even further, you might have other types of proxies, like maybe you're measuring pH, which often is driven by what the primary producers of phytoplankton are doing. Or maybe you're measuring an impact, such as dissolved oxygen, and you see a dissolved oxygen crash, and so you suspect that something out there is a bloom has crashed or something. So we'll kind of be working through different parameters and tools that will land in different parts of these, in different rings here, and then they can get stitched together. All right, next slide. And so an example of this comes from the freshwater harmful algal bloom strategy that Jamie Smith shared a little bit about yesterday. This top figure is a way that these different tools were pieced together. So this was thinking about swimmable uses, so recreational impacts. And in that box number 1, it was, well, what would you need to do to determine if cyanobacteria might just be suspected to be present? And then in that report, there was a number of parameters that were then listed out that would be relevant types of data that would at least help you determine, is there the potential for a lot of cyanobacteria to be here? And then you move over to box 2, where you would then say, well, now we want to actually know if there's toxins present. And so what types of measurements would we need to know to actually confirm that there's toxins present? And then there's also then an additional step of actually quantifying the toxins, at which point, once they're quantified, you could then get to step 4, which in this context was trying to issue a health advisory or a public health recommendation. And so you can see here how there's a lot of parameters that have been woven together in somewhat of a tiered framework, depending on the different management questions and needs. And then in the bottom left, where it says associated water quality measurements, those are water quality measurements that are more getting at questions related to drivers. So like, what's causing the cyanobacteria to be here? So you might not have drivers questions if you're just trying to make a health advisory question or posting and decision, but you might be interested in drivers if you're trying to prevent future blooms from occurring. And then in the right is one of the tables from the report, which shows for each of these indicator groups, which are sort of similar to the concentric rings on that diagram I just showed, what types of actual parameters and measurements that sort of get you information related to that group. All right, next slide. So I'm going to go through a bunch of different types of categories of tools and techniques here, and again, kind of just be going through the list with the hopes that this might help inform some of the conversations we're going to have throughout the day. And also, I am going to be mentioning several brands and manufacturers. This is not meant to be a recommendation. It's also not meant to be exhaustive of everything that's out there. This is just up there. All right, next slide. So first, just a quick overview on toxins. This is often what we're concerned about. Cyanotoxins are molecules that are harmful to humans and animals, and they're very diverse. And so on the left there, you've got a list of several of the main toxin classes and what organs they affect. And then on the right, you can see the molecular structures. And the main takeaway here is that these are different molecules with different chemical characteristics, different toxicities. And so it's there aren't really methods that necessarily just measure everything. You usually have to have targeted methods designed, or you need to choose which analytes you're going to be measuring and what are the things that you care about. Okay, next slide. So that being said, there are a number of rapid cyanotoxin tests now on the market. There are these Abraxas strip tests that Spencer Fern talked about yesterday, where you can dip that strip into the water and a bar will form. It's kind of similar to like the COVID rapid tests. They are somewhat semi-quantitative and they have a relatively low saturation threshold, only about to 10 micrograms per liter. But they're pretty cheap and they're a great way to be screening water bodies and a great resource if you don't have a big budget. There's also a new, rather newer company called LightDeck that has a fluorescent assay that can test for both microcystins and spermopsins with the results relatively quickly. I actually haven't used this one, but I believe Central Valley Board has one. Yep. So, and Ted, do you have one too? Yeah, and that's what I thought. Yeah, so you can talk to Janice or Meredith or Ted for information about that. But these are some of the rapid tools that are available. Slide. And then when we move to the laboratory, there's two main methods for measuring cyanotoxins. The first is ELISA. What this does is it's looking for a common molecular structure and then measuring that in a water sample. So in the case of microcystins, you can see the molecule up there. All microcystins, even though there's hundreds of variants, they all have that ADA amino acid arm. And so you do an ELISA in a 96-well plate, and when you buy the kit from the manufacturer, there are special antibodies that are designed to bind to that ADA amino acid arm of the molecule. And so when you put your sample in, if there's microcystins, they'll bind to that, and then a color reaction will occur, and the change in color is proportional to how much microcystin you have. These are relatively easy to set up in a laboratory. However, the downside of them is you can't discriminate between cyanotoxin variants. So even though there's hundreds of microcystins, you don't actually know which ones are in your sample. You just get this total number result. And then there's also can be more issues with matrix effects and cross reactions, because if there's anything else that has an ADA arm in it, it will also bind, and you might get false positives or could also have interferences. Slide. The other method is liquid chromatography and mass spectrometry. This uses a much more expensive and complicated analytical instrument that often requires more training. But what it can do is it can actually separate out the different types of toxins. And so this figure here sort of shows like a example of what the data looks like, but each one of those peaks there is a specific molecule that you've been able to isolate in the analytical measurement. So you can now see of the hundreds of microcystins, which ones you actually have and how much of that you have. It is more expensive to purchase and run one of these, but you get a lot more information. The downside is you really can only quantify the molecules that we actually have standards for and that there's been research done. So don't always get the full picture. And as a result of that, the results for ELISA are not always directly comparable. Go to the next. An example of that is this is some data that was actually presented at the CyanoHABs symposium a couple weeks ago from US EPA, and they were kind enough to share their chart with me. But this shows their measurements. The ELISA is on the x-axis. Actually, you can't see that. And then the LCMS is on the y-axis. And you can see there's a lot of points below the 1-to-1 line. And so that means you're getting higher ELISA detections than you are LCMS detections on this sample. And this is a relatively common pattern that you see with data. Slide. Another method for measuring toxins are spat samplers, and Meredith Howard talked about this yesterday. So often when we're measuring samples, we just grab a sample of water, so just fill a bottle up and then the toxins in it. Spat samplers are a passive sampler, so there's an adsorption resin where dissolved toxins in the water column will stick onto that resin. These are very effective at concentrating toxins over time, which makes them very sensitive. So you often will get spat detections when you're getting non-detects the water samples that you're collecting. And so it's effective to really getting the full picture of what is actually out there in the environment, what types of potential might I have, even if it's not really like a high level right now. The downsides are it is semi-quantitative because we don't always know the exact kinetics of how they're absorbing and desorbing. And so it's not easily compared with the grab samples, and there's no real regulatory framework or like thresholds for this. So it's challenging to based on spat data. And we are using spat samplers in the Delta right now. We have a bunch of DWR spats. Next slide. Okay, so now we're going to kind of move away from toxins and talk a little bit about in situ sensors for HABs. And so we'll kind of be moving more to like the algae, phytoplankton, and proxies zone of our diagram. Slide. So I think a lot of folks are familiar with chlorophyll or phycocyanin sensors. There's a large network of these in the Delta and the Bay. The principle is in this figure on the bottom right that as part of the photosynthetic apparatus, there's a lot of molecules that interact with light. And so if you can design a sensor that targets the absorption or the fluorescence of those different pigment molecules, you can estimate the biomass of that organism in the environment. And chlorophyll will give you sort of total algal biomass or abundance. Phycocyanin is a molecule that's more used for cyanobacteria, so you can use that sensor to more target the cyanobacteria. Next slide. And more recently, there's been tools that will actually get you more of the community composition. And so what they're doing is they're taking advantage of the fact that different algal groups will use different combinations of those pigment molecules, and you can kind of separate them out. And so we've been using a device called the fluoroprobe, and that separates into green algae, cyanobacteria, diatoms, and cryptophytes. And there's a little time series of what the data looks like, where you can see, in this case, over those days, you had green algae, diatoms, and cryptophytes in the sample, but not much blue-green algae. And the USGS has some of these deployed, as well as EMP uses some Sentinel boat as well. All right, next slide. And then the environmental sample processor was brought up yesterday by Peggy Lehman. This is what was called like a lab in a can, but it's a way to do in situ toxin testing. It's very expensive. It's basically like running that whole ELISA analysis in a can underwater. But this was developed in a marine environment, but there are 2 of these deployed in Lake Erie. And there's some screenshots from the Lake Erie dashboard where it will collect a water sample, conduct an ELISA, measure, get a cyanotoxin concentration, and then send that back via satellites to the mainland. And then they've got a dashboard with a daily time series of concentrations in Lake Erie. All right, so now we move over to the next topic, remote sensing as a way of monitoring HABs. Next slide. So remote sensing is basically anytime you're measuring light reflected from Earth, and there's kind of the 2 main ways to 2 variables are both the spectral resolution and the spatial resolution. So how many wavelengths of light are you measuring, and how big is each of your pixels? On the right there is showing the full spectrum of light, and then each of those vertical bands is or bars is one of the bands that one of the satellites is measuring, and that would be called a multispectral sensor. We've got like 10 to 20 measurements. Hyperspectral is something that more and more data or sensors are coming out at the hyperspectral capacity, and that's where you're basically measuring like every 3 nanometers. So instead of 10 or 20 bands, you've got like 200 bands of data coming in. You get a lot more information. There are already algorithms for estimating cyanobacteria specifically, or also just overall chlorophyll. But when you're working with remote sensing, you're not going to get toxin data from remote sensing. And here in the Delta, we are currently using data from the CYAN project, which is an interagency remote sensing project focused on cyanobacteria. And you can see there in the bottom left a screenshot from the website that the water boards funded San Francisco Estuary Institute to design a custom web interface to take that CYAN data, visualize it for the state of California. The downside with that is that it's a 300-meter pixel resolution, so we don't get some of the smaller networks or channels in the Delta. There's another satellite called Sentinel-2, which has more like 20-meter resolution. There's no operational products for Sentinel-2 for HABs, but there has been a lot of research done on it. So in the bottom right is a project that Rafe Cadella at UC Santa Cruz and SFEI have been working on, and that's looking at Discovery Bay. And you can see how with those smaller pixels, you can actually start to resolve that variation within even just an area like Discovery Bay and see which one of the little coves has 4 or less chlorophyll. Next slide. And then other like more cutting edge remote sensing applications that are being explored is you can put them on both airplanes and crewed aircraft, uncrewed aircraft or drones. Another area of exploration. You can also put them as just fixed stations. And so in the bottom left is a project we have in the Delaware River, where we've got a hyperspectral sensor. And what people are trying to do is see if you can use these sensors like you would use like a YSI XO or like an in situ underwater sensor. So we were able to get a pretty good correlation between the hyperspectral data along with the chlorophyll data from the YSI. And then the ultimate goal is that the hyperspectral data might let you actually start to distinguish between different types of phytoplankton. And so the bottom right is from a recent paper from some other USGS colleagues, or on Upper Klamath Lake in Oregon, they were able to take satellite imagery and identify which type of cyanobacteria was blooming in different portions of the lake. And so this is really an exciting application for the hyperspectral, and it could really transform our ability to track phytoplankton over time. All right, next slide. There's also a lot of autonomous vehicles that are also being developed. So where you can sort of put a underwater vehicle or surface vehicles. There are also now a couple sailboat companies out there, and a lot of this work, there's several projects in Lake Erie and Lake Okeechobee exploring these opportunities where you can then instrument these vehicles with a number of sensors and just deploy them and get a lot of spatial data and flow data. Next slide. So visual observations, new category. Next slide. So there are a number of apps and crowdsourcing opportunities for tracking blooms. Fortunately, these occur. They're often conspicuous. They change the water color. So the Bloom Watch app was developed by US EPA up in New England. And it's an app where if a citizen or anybody sees a bloom, they can make observations, and that will then send it to the relevant agency. Also here, the State Water Board is developing some work with the Water Reporter app, which Gearheart is going to talk about in a couple talks. So I'm excited to hear more about that project. Next slide. And then there's also the Microcystis Index, and this was developed by DWR, by Peggy Lehman, who's here in the room, and it's a visual scale of just how much cyanobacteria or microcystis do you see in the water. And this is now being adopted by other entities in the Delta. So we're doing it, USGS is doing it when we're out in the field, as well as some other agencies, and it's starting to turn into one of the most spatially comprehensive data sets that we have of cyanobacteria in the Delta, even though there are definitely issues with observer bias and variation, but because it's free, I mean, it's cheap, fast, and easy to do, it does seem like it's been something that other agencies have been willing to add onto their SOPs due to its ease. Okay, next category is molecular methods. So the key idea with the molecular work is that toxin production is controlled by genes. So instead of looking at the toxin molecule, you can also just look at the genetic potential for toxin production. So you don't actually know if toxins are being produced, but at least you're knowing if the cyanobacteria that are there are capable of producing toxins versus if they're not capable of producing toxins. Next slide. And so one method for this is qPCR, and the advantage of these molecular methods is that they're cheaper than the toxin analysis methods, and they're also very sensitive. So you can often get qPCR detections before you get ELISA detections, and it's cheaper. But the downside is you don't actually have the actual toxin result with you. Next slide. Another molecular method is the barcoding. And so what you can also do is look at the actual sequence of DNA in your sample, and each taxa will have a unique sequence. And so then you can get community composition data from your DNA data, and this could be an alternative to microscopy. Next slide. All right, our last category is microscopy. And so this was sort of the tried and true method for centuries, where you put your sample under a microscope and look at the different morphological variation of your taxa, and then you can count them. But it's costly, and you need some expert taxonomists, and it can take time. Next slide. So ways people are trying to speed that up are based off of flow cytometry. So here, what you're doing is you're taking your sample, you're using microfluidics to put it into a system that will then separate out the particles and use a microscope camera to take pictures of each particle. And then you can use machine learning algorithms to classify the type of particle it is, the type of cyanobacterial cell or algal cell based on its shape and look. And so it requires a fair bit of infrastructure to set it up, but once you've done that, you can much more rapidly and quickly count your samples and if you're microscopy. Next. The imaging flow cytobot is a sort of a field version of that, where you can now deploy this on a pier or underwater, and be collecting these data in real time and sending it back to the lab. It's sort of like that ESP. And this is being done with that CalHAB program, has several of these on the coast of California. And Rafe Kudell has been putting this on the USGS monitoring cruises that go from the lower South Bay up in the Delta. So we do have some IFCP data being collected. And then my last one here is community science microscopy. So as we mentioned, community scientists can be trained to learn taxonomic identification. NOAA has a program, US EPA has a program. I also want to mention there's some private public efforts to use artificial intelligence for microscopic identification. And there's a project in New York where they gave out microscopes to community science groups to collect thousands of pictures. And now they're using AI to try and automate the identification process. Patrick Sanders has a version of the field microscope that they were using for that project. And it's one of the newer field microscopes that has some really easy interface scientists. Talk to Patrick if you want to see that. He brought that here today. All right, next slide. So in summary, of course, the goal is, what are the goals? I kind of gave out a really big palette of tools that you can use. There's a lot of resources in the FHAB monitoring strategy. And so I think I'm out of time, going quick. So hopefully this is helpful for everybody.
Yeah, we have some time. You want to do an in-person one first? Looks like Sean has it.
Thank you for that comprehensive. It was very interesting. I was wondering your thoughts on the visual on the field. It's adopted by most other agencies, but that bias on the 2-3 score is really bad. So does it matter that it overlaps so much on the scoring because maybe it doesn't change management's decisions, so it doesn't need to be better, or do you feel that it does need to be refined to be much more accurate? Is it fine the way it is, or does it need to be more accurate?
No, there's room for improvement. Thank you. Yes. So the question was with the microcystis index, is it fine the way it is? Is there room for improvement? How consequential are the observer biases and variation in different programs data? I think there's room for improvement. It would be great if like a calibration day. Yeah, workshop of folks who are doing this, get folks out in the field together and start to be sharing. Yeah, have more of a sense of how we're calibrating. But I've been encouraged, even with that bias. I think just the level of detection, just going from non-detect to detect, less concerned about something like that, and even that is just that's important information having. So I think there's ways to aggregate or just because it's absence and not use all 5 levels that helps smooth over server bias. But it'd be great to be able to come together and do more. Oh yeah, Peggy has a.
I'd like to add that.
Yeah.
It's an important thing that index is developed really for microsystem. Today we don't have just microsystem. So Keith and others have been working on. We think that we need to develop indices beyond microsystem. Yeah,
I'll repeat that. Yeah, thanks, Peggy. I overlooked that. Yeah, so Peggy's comment was also that this was developed for microcystis, and now we have more species in Delta that are being observed. And so, yeah, I agree. That is another area we need to come together and get consensus on how we're handling sort of spermophanosauron or other effects of how that moves. Thank you.
Thanks, Keith. That was really helpful. Our next presenter is Rosemary Hartman from the California Department of Water Resources, and I'll let her introduce her talk.
Sorry about that. Door rang just at the wrong moment. Hi, everyone. My name is Rosemary Hartman, and I'm from the Department of Water Resources, and I'm here to talk about our effort to really use some of the different types of data that Keith was highlighting And I've called this harder, better, faster, stronger, because I really feel that putting all these different types of data together really does allow us to say a lot more than perhaps we thought we could about harmful algal blooms, even despite our lack of having a cohesive program. I'm going to try and concentrate just on the data integration efforts, not as much on the actual analysis and outcomes of a report, but happy to take questions, and the whole report is available on our the State Water Board's website. Next slide. So the great part about this project was the number of different people who contributed data, who helped us review, who helped analyze. It was really a group effort and showed us how much we can do when we all work together in an organized way. Next. So, as I'm sure you all know, we are in a drought, and we have been in a drought for a while. And in the beginning of 2021, coming out of the 2019-2020 water year, things were dry, reservoir levels were low. We thought we were going to be in okay shape at first, and then quickly realized that we were not. We were in a very severe drought. The projected runoff did not come out the way we had expected, and we needed to do a TUCP, or temporary urgency change petition, and a emergency drought barrier in the Delta in order to maintain water quality. Next slide. So that temporary urgency change order that was granted from our petition reduced our export requirements in June and July from 4,000 to 3,000 cubic feet per second. And during that time period, project exports were reduced to below 1,500 cubic feet per second. Next slide. The emergency drought barrier was put in West Falls River right next to Frank's Tract. This was installed in the end of June 2021. It was actually just, well, it's in the process of being removed right now. It was just breached last week and will be totally removed by the end of the month. Unlike the 2015 barrier, which was only put in for this summer, this actually stayed in all through last winter. It was just a notch put in there for fish passage and water to flow through, and it was filled in again in April, I believe. Next slide. So there was concerns with putting in this TUCP and urgency drought barrier that harmful algal blooms might get worse in the Delta. Now, when we're trying to look for the effect of a management action on any particular thing, as a scientist, I like coming up with a before-after control impact design. That's kind of the rule of thumb for how you test the effect of an action. Is you look before and after the action, and then you look in places with an action and places without an action. Unfortunately, doing before after was going to be hard because we were having to do this analysis before the barrier was taken out, so we didn't have like totally after. And we were told that we would have to do this like right before the barrier went in, so we couldn't collect a lot of before data. We're going to have to rely on existing data. However, we could do some comparisons interannually, so we could do year-to-year comparisons. Looking at previous dry years without a barrier, like 2014, 2016, 2020, previous dry years with a barrier, 2015 and 2021, and previous wet years, 2017 and 2019. And we'd expect if there wasn't an effect of the barrier, that years with a barrier would look pretty much the same as dry years. There's going to always be a little noise. I should have put error bars on this. This is just sort of a fake mock-up graph. There's always going to be some noise. Some years might be a little higher, a little lower. You kind of got to look across all of your data. But if there was an effect of the barrier and TUCP, you'd expect a consistent difference between a normal dry year and a dry year with a barrier. Now, unfortunately, dry years with a barrier are always going to be drier than regular dry years because there was a reason we needed that barrier and that TUCP. We didn't have enough water to go without it. So it's going to be different than other previous dry years. Next slide. While we don't have a control delta, we could do a regional analysis to see which regions are more likely to be affected. The Cache Liberty area, we didn't expect an effect because it's mostly tidally influenced. Upper Sacramento, increased residence time due to reduced flow. Lower Sacramento, increased residence time, increased salinity. Lower San Joaquin, increased salinity, and a change in velocity due to that barrier. The Old Middle River and Franks Tract areas, velocities would change, salinity would go down, and residence time would go up. We didn't expect any change in the South Delta or East Delta because they're not along the Sacramento River where we had the outflow change. They're not super influenced by the reduction in exports, and we didn't have any changes in the San Joaquin side. Next slide. So making these comparisons, we can compare regionally and to previous years, but we don't have a HAB monitoring program. So how are we going to compare this year's data to previous year's data if there's no data? Well, there's a lot more data than we really would have expected. It's true we don't have an integrated monitoring program focused on CyanoHABs, but a lot of our existing monitoring can be leveraged to tackle these questions. And what I really learned through this process is we have a lot more, we can say a lot more than we thought we could, but there's still a lot left to do. And I'm glad I'm going right after Keith because I'm going to highlight a lot of the data sources that he was talking about. Next slide. So those visual assessments, which there's been some talk in the chat and after of like, well, how accurate are these? There is definitely a lot of variability between surveys. I'd love to see, as Keith was suggesting, a intercalibration day where we all look at buckets of water with different amounts of microcystis in it and calibrate each other on what's a 2 and what's a 5. But we have great spatial and temporal coverage. Thousands of these data points come in every year at this point. And after I put together our data set, I learned that 2 other programs had just started doing this. So what it lacks in precision, it makes up for in volume of data. And we can at least get presence, absence, low, medium, high, order of magnitude kind of deal of where the microcystis is, how frequently we're seeing it, and how bad is it. Now, as I mentioned, variability between surveys, and it's only really good for microcystis, not some of the other cyanobacteria. People are starting to use it for F-anisomenon as well. That's less common. Next slide. So what we see is that we get more microcystis in dry years. Those are the ones in red. This is just the relative frequency that we see these, on a score of 1 through 5. The pale blue is when it's absent, and you see it's more likely to be present in those dry years. Wet years like 2017 and 2019, less likely. But of note is that 2020 and 2021 were not statistically different. And 2015, 2016, 2014 were not statistically different. We didn't see a difference in years with and without a barrier. There were some challenges in getting all of these data put together. Fortunately, the Interagency Ecological Program Synthesis Team, particularly Sam Bashevkin, who's now with the State Water Board, has done a lot of work to get some of the long-term monitoring data integrated into integrated databases so that all of them are in one place. But that is usually after all the data has been QC'd and published, it's then integrated and put together. What we had to do is, call the primary investigators, hey, can we please get a preview of your harmful algal bloom index data? It's not normally reported right away because it's more for long-term status and trends. So that took quite a bit of work just to round up all the data, but we ended up being able to say things like this with it. Next slide. The other really useful thing is other data is collected along with this data, in particular water clarity, Secchi depth, temperature, salinity, other things. We looked at a number of different parameters we thought might be useful predictors for microcystis occurrences in the South Delta. Just using these visual indexes, And we were able to create a pretty good model of the frequency of low or high microcystis occurrence based on project exports, Secchi depth, which is a measure of water clarity, and temperature. We found increased project exports decreased the probability of microcystis, increased water clarity, increased probability of microcystis, increased temperature, increased microcystis. Next slide. Satellite imagery that Keith mentioned has been great. We have this continuous record over the course of the season. Good spatial coverage over the entire delta. It doesn't tell us what kind of cyanobacteria. It's also not sensitive at the low end of the range. Can't pick up small amounts. And you can't see into small sloughs, but it is really good for looking to see what's happening right now. Is there a bloom going on right now? And mapping the course of a bloom. Next slide. We saw a bloom in Frank's Tract in July and August of 2021 that started kind of on the eastern edge of the tract and then moved over the entire tract. Next slide. We also could use continuous sondes that give you a record over the entire time period for multiple parameters, not just chlorophyll, but turbidity, temperature, etc., pH, dissolved oxygen. They're only in certain locations and they're not going to give you toxins or community composition. But we found they're very useful, especially for from the effects of harmful algal blooms. Next slide. In particular, we saw that Frank's Tract had very high pH due to all of that photosynthesis going on. This is the top slide there is pH at Frank's Tract over the years. It always gets high in the summer, but it got particularly high in 2021. We saw that bloom. And we had particularly high dissolved oxygen in 2020 as well. Next slide. We can also take those same continuous probes and instead of putting them out in one place, use our boats to take continuous water samples throughout the delta and get a map in the end. Multiple parameters, you can pair grab samples for lab results with your high-speed mapping, but they are limited in temporal scope. Only, about once a month can we do this. And we do have a fluoroprobe, which gives us those community classifications, but it's not going to give you exact taxonomic resolution unless you take a grab sample for further analysis later. Next slide. And so we basically were able to validate that satellite data with our boat-based mapping surveys. We saw this bloom in Frank's Tract and the surrounding sloughs. We were able to get into those narrow sloughs that the satellite missed and find very high levels of cyanobacteria around Frank's Tract and Old River. Next slide. Toxin analysis. And this is, of course, the center of Keith's circles. This is what we really care about, is the impact these cyanobacteria are having on humans and the environment. This is the one place where I felt like in putting together this data set, we were really hampered in being able to say the impacts of the barrier because we didn't have comparisons to previous years. There was bits and pieces of toxin data that have been collected in previous years, but it wasn't in a consistent enough manner for us to really make good comparisons at most sites. There were, a few areas where we could do that. Like there have been continuous toxin analysis and for the State Water Project for a long time, at Big Break Regional Shoreline. Certain pockets of it have definitely been monitored well, but it was difficult to say things about what was going on in the Delta as a whole. Next slide. What we saw, based on that areas of sort of the open water in the Central and South Delta and even the North Delta, was toxin levels were relatively low where we had data. The bloom in Frank's Tract that looked really bad on the satellite, the only toxin analyses we had said that it was just barely above the caution level. There's other spots in the central and south delta on the lower San Joaquin and Old Middle River where it got up to 2 or 3 micrograms per liter, but not above the warning tier. Now, there were some of the pockets in the delta like Discovery Bay, Stockton, had much, much higher levels, but the open waters were relatively low. Next slide. We also put together the community composition data that we've already talked quite a bit about in these workshops in terms of it's great for knowing exactly what species are present, but there are issues with different labs doing things differently. It would be great if DNA analysis was a routine part of this. It hasn't quite got there yet. Also, one of the things that hampered us in putting this data together is most of our community composition data came from the environmental monitoring program, and it's taken at 1 meter depth. Now, microcystis and many other cyanobacteria form surface scums, so a sample at 1 meter isn't telling you much about what's happening in that surface scum. Next. What we did see, in looking back at EMP's data over the past, 8 or 10 years, is that microcystis is one of the most common cyanobacteria we see, followed by Athanazoa. But Dolichospermum is also present and was particularly high in 2021. I don't have the 2022 data on here yet, but we actually saw a huge Dolichospermum bloom in Frank Truck in 2022, and that was really surprising. It's something we hadn't seen before, really. It wasn't toxic, but there was a lot of Dolichospermum in Frank's tract, and we're still not sure what to make of that. The other thing we really saw in putting this together was that it did not match visual observations very well at all. And since this report, Ted Flynn has done a little work looking at surface tows versus samples from 1 meter depth. You have a much different relative composition of cyanobacteria versus others. In 2020, there were almost no microcystis seen in any of the grab samples, but visually people were reporting it all over the delta all summer. So there's definitely some issues there. There have also been some challenges with this particular data type in putting together data from multiple labs, especially if they use different magnifications. They have different taxonomists putting things together. Even at the genus level, sometimes there'll be very little overlap between labs. Next slide. There's also a lot of tools that we can use in terms of modeling. The hydrodynamic modeling that we used for this report allows us to assess hydrodynamic and hydrologic drivers like residence time, water temperature, salinity. There's some work being done to actually do biogeochemical modeling to try and predict when we would get blooms of phytoplankton or cyanobacteria. Those are still in the works. Now, in doing these residence time models, there are still issues with garbage in, garbage out. You need to make sure you have really good data in terms of bathymetry, water sources, hydrodynamics to parameterize these models. And one of the particular problems is that vegetation can really impact where water is flowing. And we don't always have great vegetation data in the right format to be used in these models. Also, different models will give you different results. In particular, looking at residence time, DSM-2, which is one very common one used for a lot of hydrodynamic modeling in the delta, is not very good at large open bodies of water. It'll give you strange numbers for residence time in those sorts of situations. Whereas a three-dimensional model like Schism can be a little better at modeling those more accurately. Next slide. So we used the Schism model to get a water age, which is similar to residence time for Frank's track with the barrier and without the barrier. And what you see is, not surprisingly, the barrier slows water down. and you get a much longer water, much older water, longer residence time in the western side of the tract than the eastern side of the tract. Now, this was a little odd because, as you remember, the eastern side of the tract was where the large harmful algal blooms started. This could have been an interaction with the vegetation that was all on the western side of the tract. It could have been that cyanobacteria, the San Joaquin, were coming in and feeding the area in Frank's tract rather than in situ production. A lot of possible theories. And I think this is one type of data that is not well distributed and not well shared yet, or at least I have not been in the right groups to know how to make this data more available and more widely used. Because this is something a lot of people want, but a lot of people don't quite understand how to use it and the best forums to find it and share it. Next slide. So incident reports, we've talked quite a bit about this already this week. Anyone can submit these incident reports, which is great for community engagement, especially in areas where we don't have routine water quality monitoring. But people who make these reports may or may not be reliable. They may or may not be accompanied by toxin data. One issue that was brought up yesterday is someone goes and reports a horrible harmful algal bloom that doesn't follow up and say, okay, the bloom's over. And the way the database is currently set up, it's really set up for us to understand what's going on right now so that people can go and see where there is danger. It is a little harder to pull the data from the entire summer and track what the incident advisory levels were over the course of the summer and how that's changed over time. Next slide. But we were, with the help of Karen Atkins from the regional board, able to pull the incident reports and map out what we were seeing. And there were definitely many hotspots, backwater areas of the Delta. We saw very high microcystin levels. But because those have been occurring every year, we made the assessment that it was probably not caused by our TUCO and barrier. Next slide. I'm gonna click, yeah. So all these sources of data weren't originally designed to test our question of interest. However, when we put them together, we really could say a lot about the status and trends of harmful algal blooms in the Delta. And we could even test our specific hypotheses about the effect of our management action. We found that the barrier probably did exacerbate that harmful algal bloom in Frank's tract, but we didn't see evidence of an impact of the TUCP on the Delta as a whole based on our regional and interannual comparisons. But we did identify many remaining unknowns. Next slide. So the next steps with putting these together from the point of view of what I would need to do a report like this again is getting regular toxin data collection, regardless of whether bloom is occurring, have it on a regular basis as part of our water quality, standardize that visual assessment index, get everyone in a room to compare notes. All same with microscopy data, see if we can work to get that more standardized, increasing use of new tools, and starting this process of figuring out who has what data where and being able to put it together in a easier and faster way, ideally on a shared platform. And with that, I will take any questions. And again, sorry for the delay.
Thanks, Rosie. That was a great presentation. I think we don't have time for questions. We're going to move on to our next question, our next speaker. But if you have questions for Rosemary, she is great to email at rosemaryhartman@water.ca.gov, and or she'll probably respond in the chat as well. So thanks again, Rosie. Our next speaker is going to be Haley Plaas, along with Hans Paerl from the University of North Carolina. to talk about aerosolized CyanoHABs and nutrient limitation. And let's stay with 1 moment as we pull it up here. Haley's joining us remotely.
Yep, ready when you guys are.
Oh, okay, you are there. Sorry, it's on our end, trying to pull up your slides. So
Awesome. Okay, are we good to go? All right, I'll jump into it. Hi, everyone. My name is Haley. I am a public health PhD candidate working with Hans Paerl at UNC's Institute of Marine Sciences. And today we're really excited to present our work on the aerosolization of cyanobacterial cells and their toxins in the Delta. And then about halfway through this presentation, Hans is going to take over and talk about our nutrient addition experimentation that is ongoing at the same time as all of our aerosol sampling that started this past summer. Next slide, please. So first, I want to give some background on how harmful cyanobacteria and their metabolites can get into the air in the first place. So this figure on the slide is from mine and Hans's review article that was published in ESNT in 2021. And this was exploring all that we currently knew about the aerosolization of cyanobacterial cells and their toxins specifically. So the thing that gets these toxins into the air is any sort of physical disturbance or mechanical agitation from wind action and wave action and even recreational activity that causes bubbles to get trapped beneath the water's surface and then burst. And that bursting emits a spray of these microscopic aerosols, which can carry those toxins and cells and other metabolites. So microcystins and anatoxins and nodularin are the 3 cyanotoxins that have been quantified in aerosol to date. But we're interested in learning more about the size fraction of aerosols that carry these toxins to better inform public health risk assessments surrounding the inhalation of these toxins. And we're also interested in finding out more about what environmental conditions, what water chemistry conditions are further promoting the aerosolization of these toxins. And then beyond that, we're also interested in looking at other associations between the blooms and air quality in general, because it's possible that other metabolites, such as these taste and odor compounds or other metabolites that are less studied, are also being emitted from these blooms and impacting aerosol concentrations in the airshed of the Delta and beyond. And of course, this all can be impacting human respiratory health because an inhalation of an aerosol particle can be toxic to human health, regardless of what it's composed of. So next slide, please. So just to present a little bit of background findings from our studies in North Carolina that have informed some of our work here in the Delta. So in 2020, we conducted very similar research in the Chowan River, which is a eutrophic estuary system in eastern North Carolina. And we were looking for microcystin and cyanobacterial cells in PM2.5, which is particulate matter with 2.5 microns in diameter or lesser. And during this study, we did not quantify any microcystin in PM2.5, and we think that this is because the main bloom that we saw was Delicospermum, and it didn't produce much microcystin at all during our study. So there wasn't much toxin in the water to begin with. However, what we did find that was very interesting is that several of these aquatic bloom formers were detectable in PM2.5, and specifically Aphanizomenon, Microcystis, and Delicospermum all increased in abundance in PM2.5 at the same time, suggesting that some environmental condition was promoting their aerosolization at the same time. And also what was really interesting is that we found a strong association between ambient concentrations of PM2.5 and the abundance of Delicospermum in all of our water samples. So this suggests that something specifically was coming off of this Delicospermum bloom, contributing to increased aerosol concentrations in the airshed of the Chowan. And we're not exactly sure what caused this, although we do know that it wasn't likely the cells themselves or the toxins, since we didn't also quantify those at the same time during this association. So that all has informed our study moving into California. So next slide, please. So the specific aims of our work in the Delta now are first quantifying 3 different cyanotoxins in both PM2.5 and PM10. So 2 different size fractions of aerosol with specific health implications when inhaled. And then secondly, we're hoping to also identify more species of cyanobacteria that are being emitted in aerosol using some microbial approaches that both Keith and Rosie explored. And then thirdly, we're going to use a series of multivariate statistical analyses to look at various environmental drivers and how those might be influencing toxicity in our aerosol samples. Next slide. So in order to sample these toxins in the air, we installed 2 high-volume aerosol samplers at 2 sites in the Bay Delta. That was Discovery Bay and then the Port of Stockton, close to downtown. So these large aerosol samplers operate like large air conditioning filters. So they pull this particulate matter across a filter, specifically quartz fiber filters, which then we can analyze offline using liquid chromatography coupled with mass spectrometry. So we're looking for those 3 different toxins and also various congeners of these toxins and these 2 different size fractions of aerosol. So again, these 2 different size fractions are going to give us more information on potential health risks with inhaling these toxins. But what we're also hypothesizing is that we're going to see more intact cells impacted in our PM10 samples, and we're going to see more of the dissolved toxins in our PM2.5 samples. So we're trying to differentiate between toxins being aerosolized in the dissolved form and then in the intact cell form. So again, giving us more information on what the potential health risk could be when inhaling, but also how far these particles can travel based on the size of that aerosol particle. Next slide. And then for our objective 2, to sample our DNA in the air, we're using these smaller low flow impinger apparatuses, which is pictured in the middle. It's that little glass thing. So these allow us to sample aerosol directly into a liquid media, which then we can use for a variety of microbial analyses, including flow cytometry. We're actually going to culture some of those samples for potential metagenomic analyses once we have enough biomass. And then we're also going to get some 16S data on that to look at various bacterial groups present in our aerosol samples. And then lastly, to answer our 3rd objective, we're also gathering environmental data from water sampling weekly while our aerosol samplers are running, and we'll be downloading local weather and PM data, which we will put into that multivariate statistical framework in order to kind of tease apart which environmental drivers are most associated with these increased toxins in the air. So next slide. So that's a really, really quick overview of our aerosol sampling that started this past summer and will go through next summer. We ran our samplers from June to September of 2022, and we're planning on April through August of next year. But with that, I'm happy to answer any questions at the end of this presentation on the aerosol specific side of things, but I'm going to hand it over to Hans at this point to talk about our nutrient addition bioassays.
Hi there, everybody. Can you hear me okay?
Yep, we can hear you.
Okay, great. Well, this is the nutrient part of the story. And I just wanted to emphasize that our project actually didn't really get started until formally until the 1st of July. So you're going to be looking at some hot off the press data from bioassays that we conducted in parallel with the aerosol work. And this is just a schematic showing you the additions that we did in the bioassay, one in June. And you can see we had controls, obviously, nitrate additions, phosphate additions, and N and P together. And then in September, we got a little fancier. We did nitrate, but also looked at ammonium, phosphate, nitrate, and phosphate, and ammonium and phosphate. And if I could have the next slide, please. Okay, so these are results from the pigment work that we did on the bioassay. So this was done by HPLC. It's basically a method for looking at multiple pigments, including chlorophyll A. So the upper one is chlorophyll A, and then we go down the road here with all different diagnostic pigments for the different algae. And this is for Discovery Bay, and you can see over a 5-day period that the algal biomass response was greatest in response to nitrogen additions in Discovery Bay. And if we go down the line here, you can see if you look at cyanobacteria, they responded really well and this is a boo-boo in the slide, not to ammonium, but to N and P together during the course of that bioassay. So evidence for nitrogen being a stimulatory component here in the nutrient-driven blooms. Next slide. Okay, if we move over to Stockton ship channel, we get a different picture. The picture mainly is that there's no difference between the controls and nutrient additions. And so there's no apparent nutrient limitation. And what that means is that there is plenty there, and any additions of nitrogen and phosphorus are not going to be significantly different from the control. Next slide. If we go to September, it's sort of overlapping here in this slide. But if we go to September in Discovery Bay, we see that the algal biomass again responds to nitrogen additions. No significant response really with phosphorus, which is all the way over on the right-hand side there in that upper line of graphs. And I wanted to point out that the cyanobacteria like nitrogen and they like N and P. So there is, again, evidence for nitrogen, but also if you add phosphorus to nitrogen, you get more than nitrogen alone. So this is starting to argue then for dual nutrient limitation, which we've seen in other places, including Lake Erie, for example, and some of our North Carolina work. Next slide. If we then go back to Stockton Ship Channel, you see again that there isn't really much of a response to nutrients, but there is a slight response, and it is significant to ammonium and nitrate. Again, no response to phosphate. And you can go down the chart there to look at cyanobacteria. You see the response to cyanobacteria is pretty strong to nitrogen, particularly nitrate, and some to ammonium as well. But really, all being said, there's really not that much of a difference between those two. They like nitrogen, and I think that's the way we should look at it. Next slide. Now, there's an interesting thing that I did just as kind of a lark during these bioassays, and that is to measure pH along with the pigment responses that we saw in the bioassays. And what I wanted to show you is that it looks like you can use pH as a bioassay response indicator. Keith already mentioned pH as being potentially useful, but you can see here in Discovery Bay in June, in the early bioassay, there is a response here to N and P. There's also a marginal response to N, and that seems to parallel the chlorophyll results that we've gotten. On the other hand, if we go to Stockton Ship Channel, the pH is pretty high to begin with, and you can see there's no difference there between the additions and controls, again indicating that there's no apparent stimulation and probably an excess of nutrients in the system. And the graph below just shows the relationship from some laboratory work between pH and the amount of chlorophyll in experimental enclosures. Next slide. If we look at September, we again see that response to ammonium and nitrate using pHs. And if you go down the line, there's not much of a significant response to P, which matches the chlorophyll response. And then N and P together also give you good response. So this again correlates pretty well with the chlorophyll assays. And the reason I bring this pH assay up is because it's really cheap to do. You know, all you need to do is buy a little field pH probe, which is this work was done with for, I think we paid $150 for it, calibrate it, and use it in parallel with our other parameters that we're seeing in the bioassays. Next slide. Okay, now we're going to just look at the ambient nutrient concentrations during these bioassays. And first thing I wanted to point out is that the axes are different for the two different places. And that's obvious because there's a lot more in Stockton Ship Channel than in Discovery Bay. But if we look at the, in this case, looking at the Discovery Bay sample, you see that there's, again, pretty low amounts of nitrogen in those samples. That would be the the bars all the way to the left there. If we look at the if we look at the response with ammonium, you see there is ammonium in the water, but again, remember that x-axis has been expanded quite a bit. So there does seem to be, evidence from the ambient nutrient concentrations that nitrogen should be most limiting in Discovery Bay, and not so in the Stockton Ship Channel. Next slide. And if we look at phosphorus, we get a somewhat similar story. Fairly low in Discovery Bay, although not low enough to be limiting, apparently, according to our bioassays. And if you look at Stockton Ship Channel, it's just astronomical. It's really high, and it also matches our results that phosphorus is not limiting in that system. Next slide. Okay, so just to sum up here, Discovery Bay, nitrogen appears to be the most limiting nutrient at various bloom stages. And looks like ammonium might stimulate best compared to nitrate, but they both stimulate it. So as Peggy had mentioned in her earlier talk, they like nitrogen in most forms that it exists, particularly if it's limiting. Phosphorus appears to be sufficient, and that's most likely due to effective internal loading, regeneration in the system. There's been loading going on in these systems for decades, what we call legacy loading, and apparently these systems operate very well on regenerated phosphorus. So you got a long ways to go to get the phosphorus out. Stockton Chip Channel, nutrients appear sufficient. There's maybe some evidence for marginal N limitation later on in the summer, but most likely the blooms are controlled by flushing and light availability. Physical factors controlling the bloom in this particular system where there's just excesses of nutrients. So overall, at both locations, and if I have time for another slide, I can show you this. Silica is another nutrient in question, of course, because it controls the growth of diatoms, but it appeared to be sufficient for supporting phytoplankton growth. So the take-home message is we need to reduce both N and P inputs for stemming eutrophication and long-term control. And that's going to be particularly challenging in the Stockton Chip Channel, which has a huge legacy of loading in it already. And that's why I stress the physical factors, flushing and light availability, as potentially being more overriding limiting factors. And the last slide, since I have a few seconds left, I think. Well, this is second to last slide. Just wanted to acknowledge all the folks we've worked with, including folks in California, North Carolina, and all the help that we've had from folks that have really helped us get set up with all this stuff, particularly Peggy. We had a summer intern there, Seyong Cheng, which did a lot of the aerosol sampling. And then folks that really helped us a lot with the aerosol sampling. Last slide. I think there is one more left in here. Oh, it got cut off. But anyway, the last slide, if you can imagine, shows very high silica concentrations in both Discovery Bay and Stockton Chip Channel. So I don't think silica is playing an important role as a limiting nutrient in those systems. And if we have time, be more than glad to answer any questions. Thank you for including me from Hurricane Central here in Beaufort, North Carolina.
There's clapping if you can't hear it, but thanks, Hans and Emily, for a great presentation. We have plenty of time for questions.
You'll have to repeat them because I'm having a hard time hearing the room.
Gotcha. Can you hear me okay? At least?
Oh, there's the silica slide. Thank you.
Oh, yeah.
That magically showed up. Okay, that's great. But you can see it's really high in both places. So no worries about silica.
Gotcha. Okay. That's what I was going to say. We have plenty of time if you wanted to add anything else. Okay. There are questions online, I guess? We have a question here about the if temperature has a threshold.
Bloom threshold.
Yeah, exactly.
Yeah.
Sorry.
Yeah, no, it does, but for microcystis, you haven't reached it. I think the hotter the better. We've actually seen blooms in North Carolina waters where it's been over 30, 32, 33, and microcystis does just well. And in fact, at those temperatures, it outcompetes almost anything else in the water, including some of the other cyanobacteria, which have slightly lower temperature thresholds. And we've also seen that in work that I've been involved in China, in Lake Taihu, where the water temperature often gets up above 30. Microcystis has the highest temperature threshold of the major players that we've talked about, including Delicospermum and Aphanosomina, for example. And in some work that Tim Otten and I did back in the Bay Delta, back in an earlier project, we also saw that microcystis really started to take off when the temperatures got up to 25 or higher. And I don't think, even with the warming that's going on in the Delta, you haven't reached a maximum yet for microcystis.
Thank you. We have a question in the room here.
Follow-up question.
Follow-up question.
Hans, have you observed any interactive effects between temperatures and nutrients?
The question was, have you
Observed interactive effects
Have-
between temperature and
have you observed any interactive effects with temperature and nutrients?
Well, yes, the answer is yes, as long as the nutrients are still available. We haven't really looked at that yet in the Bay Delta, but I can tell you from the China work on Lake Taihu, which has a very similar algal assemblage, that if you get up above 30, you can still add more nitrogen to the system and increase the bloom. So, it's just an organism that just really likes it hot. And the more you can get around the nutrient limitation issue, the better it does. And that's really a problem, of course, with global warming. The other issue, of course, is with droughts. And this gets a little off your question, but I just wanted to bring it up. With droughts and increased residence time in these systems, poor flushing, I think things could get worse. And that's one of the reasons why I stressed that yesterday in my comments, that, with a place like the Stockton Chip Channel, it may be that physical forcing is going to be the most effective way to minimize the bloom and keep it under control until we really have sort of leached the nutrients out of the system. And I realize that that's not an easy thing in California when you have protracted droughts and no water to really play around with. And then, of course, the whole issue with water use and withdrawal of water, doesn't make that any easier.
Thanks. We have another question in the room here.
I was wondering if you can expand more on that. Flushing idea for action. Around the world, nutrient loading is generally the go-to for controlling blooms. Why flushing over nutrient loading? That seems to be also part of why not do both.
Yeah, I think I heard your question. Yeah, it would be nice to do both, but I think there's also a legacy of nutrients in these systems. And that's why I mentioned the regeneration. It could take a long time to sort of leach the Stockton Chip Channel of phosphorus, for example, which has been, accumulating there for decades. So even when you reduce phosphorus, the system is still going to operate on regenerated phosphorus. We've seen that in Lake Erie, for example, in work we're doing there. So, obviously we need to reduce nutrients, but I would say in a place like Stockton Chip channel, don't expect an immediate decrease when you have all that internal nutrient loading going on at the same time. And that's why I mentioned the physical factors as being potentially important to consider as well.
All right, thank you so much. We're getting ready for a break, but thank you so much, Hans and Haley, for presenting. It was a really great.
Thank you for having us.
Yeah, of course. We're so glad you're able to join us here. So right now it's 10:20 and we're going to have a break. We'll be reconvening at 10:30. Thanks.
For the opportunity. There's a good number of people that are co-authors on this talk on the list here. Next, please. And a large number of folks involved with field work, lab work, etc. Next, please. Okay, so San Francisco Bay, as we all know, highly enriched in nitrogen, primary source from wastewater treatment plant effluent. The green circles on the map to the left are showing the area of those circles represents the mass load in kilograms per day from the roughly 40 wastewater treatment plants discharging throughout the system. Bay has been relatively resistant to its nutrient-enriched status over the years. That's been tested recently and failed. The nutrient management strategy for San Francisco Bay was established in 2012 with the key aim of asking the question, do loads to the bay result in adverse impacts either now or under future scenarios, and what management actions are needed to prevent or mitigate current or future impairment? Next, please. So there's enough nutrients in this system for us to find ourselves in the condition B, where large blooms, low dissolved oxygen, harmful algal events. We've often have found ourselves over the last several decades at condition A, low production, ample oxygen with the high suspended sediments, strong tides, strong winds, and short-lived stratification being the things that kept us in this relatively better state in terms of condition. Next, please. But a major concern has been not just monitoring chronic or routine conditions in the system, but also could the system actually shift if any of these constraints on production are relaxed. Next. And so the science focus areas for the San Francisco Bay Nutrient Management Strategy are to understand nutrient loads and transformations, phytoplankton blooms and low dissolved oxygen, and then as well, HABs and toxins have been a major focus. We've been adding gradually over the last several years, since about 2014-15, our collaborators, various techniques for tracking HABs and toxins throughout the system. Next. So to give you a quick overview of this event, in late July, the event was first observed around Alameda Oakland Deep Shipping Channel. The organism was identified as Heterosigma akashiwo, toxic to fish, and it's been on our list of watch list of organisms to be concerned about. Next. So we first started tracking this using remote sensing. We heard this event was possible. We've seen a lot of these events over the last several years start up, show a little bit of a flicker, and then peter out before there was anything needed to follow up. But we started tracking this via remote sensing. Next. And the event gradually migrated out into the deep water areas off of Alameda, and then gradually spread throughout the system, and then beginning on the 30th of August, abruptly terminated. Chlorophyll concentrations were in the range of 50 to 100 micrograms per liter throughout South Bay, south of the Bay Bridge. 20 times typical summer values. And as many of you have probably seen in the papers, fish mortality in South Bay, Central Bay, San Pablo Bay, and dissolved oxygen deficits in particular in south of the Bay Bridge. I'm not going to talk about the fish impacts, but that's obviously clearly a motivation for this work. Next. So some of the key questions that we're asking ourselves right now are, what's the likelihood of something similar occurring again in the very near term? For example, might we find ourselves in the same situation next summer? And over the next several months, is it possible for us to understand whether it would be feasible to stave off or mitigate impacts with some quick intervention? For example, reducing loads for a few days, if that was possible. Would that have an effect of stopping a bloom like this? And then next, what longer term management options would be effective at preventing something like this in the future? Next. And that essentially a high level question of what factors caused or shaped this event over time. Next. And so I was going to go through and give an overview of the event, the water quality impacts, and then some quick overview of the observations and hypotheses based on that early work, but really trying to emphasize what the data collection was over this year to fit in with this workshop. And I guess ask the question as well, is that enough, or what are the other things that we need to be doing? Next. So this is a bit of a dense slide, but showing you the monitoring and data exploration resources. So over the last several years, our program's been adding moorings throughout South Bay, not just in the deep channel, but also on the shallow shoals in South Bay. We've been working with USGS biogeochemistry team over the last several years on doing some pilot tests where we're doing this high frequency mapping out onto the shoals throughout the system. So essentially we had created the muscle memory of being able to do that work and had really nailed it down into also with the data processing. The measuring things like chlorophyll, dissolved oxygen, nitrate, pH, turbidity, fluoroprobe for phytoplankton community, as well as discrete samples, chlorophyll, nutrients, but also for collection at each of the vertices on this mocked up cruise track up here for 18S and 16S for phytoplankton community and microbial community. When possible, we did vertical profiles in the deep channel. We piloted this over the last several years, and then once we saw this event starting to take off in early August, we were fortunate that the USGS team was able to mobilize, and we were out there for a total of 6 or 7 days over a 6-week period. We also continued to have the long-term deep channel monitoring from Peterson, the remote sensing data that I mentioned before, and you can't see on this here, but right below off the screen here is numerical models, the use of numerical models for stitching this all together. Next. So this is showing that the two colorful images there are two of the remote sensing images, but then right above those are the mapping cruises showing chlorophyll concentrations during that event, and more or less reasonable correspondence between those. The remote sensing product is actually giving a reasonable estimate, or at least a good forecast of what's happening there. You can see as well the chlorophyll gradually increasing as you elevate it to the north, but very low to the south. Next. And then gradually that same gradual increase moving further to the south. Next. And now I'm popping up here one of the moorings to show you the same information that this SLH on about a 5 meter deep, 3 to 5 meter deep location, as well as the Dumbarton Bridge. And you can see as well this gradual increase in chlorophyll. Fortunately, the x-axis is cut off there, but it's around August 15th to 18th where the chlorophyll concentrations start to increase and then remain elevated. Next. And then they abruptly drop off, consistent with also with remote sensing. Next, please. So if you look at that amount of chlorophyll and the rapid decrease in the chlorophyll that happened, you can turn that chlorophyll into roughly an oxygen equivalent. If that was all degraded very quickly, how much oxygen demand would that exert? And it's on the order of 5 to 8 milligrams per liter of oxygen. So we were quite concerned when this crash was beginning to start that this would translate into low dissolved oxygen throughout the system. Next. And these are some illustration of these various locations: the San Mateo Bridge deep channel, Dumbarton Bridge deep channel, and then 3 shallow locations out on the eastern shoal, with dissolved oxygen increasing and then gradually leveling off, gradually decreasing and then plummeting. And with next, please, resulting in large regions of South Bay and Lower South Bay with dissolved oxygen less than 5 milligrams per liter for 7 to 10 days. Less than 3 milligrams per liter for 2 to 3 days over large swaths of the system, around 250 square kilometers. So when we start to get down into that range of 2 to 3 milligrams per liter per day, that's where most organisms are not going to be happy or with that, or death over a large portion of South Bay. Next. So coming back to these questions of the more mechanisms, like trying to get a sense of what factors caused or shaped a HAB event, and HAB events, as we've talked about, are notoriously difficult to figure out what exactly was the trigger. Next, please. And so I think if we break this question down, though, there are some questions that we probably can't get our heads around or really be able to get well. Like, for example, what caused the initial event in the Alameda Channel? I don't know that we have the information ahead of time to be able to predict or understand exactly what caused that. But for the subsequent steps, next, please. Things like what allowed the bloom to take off initially in the deep channel, and then what caused this? What caused the severity or contributed to the severity, the magnitude, the duration, the area? What allowed it to spread throughout the system? And then lastly, bloom termination. I think we do have a shot at trying to answer some of those, right? I sure hope so, because those are the things that we're trying to understand over the next several months to anticipate management needs. Next, please. So a conceptualization of this. So the organism, this is a very terrible drawing. I'm a scientist, not an artist here. So we've got heterosigma coming out of the Alameda area, coming into this area right offshore. What allowed it to first be able to take off in the deep waters just offshore, just to the edge of that? And then next, please. What would have contributed to it, let's say, going to the north or going to the south? We saw more of the event was definitely centered to the south. And some of the things that we need to consider here are water column depth, for example, for phytoplankton to be able to grow. We also need to understand the physics and the transport in the system. How much of it was actually tides were pumping it to the north versus pumping it in general to the south. Next. Irradiance coming in, it says both the amount of sunlight, and it happened to be that this was a period of time where there was a lot of, we had a number of clear days during the month of August, which we think may have contributed to the additional light getting down to the system in the first place, in addition to light attenuation in the water column, potential for stratification, short-lived stratification in the surface layer. Next. We need to also be thinking about vertical mixing, which would, especially when we consider the generally elevated suspended sediment concentrations, limiting light availability and the potential for growth, resuspension of those sediments in the shallow areas, both by tides and wind. Next. And then next is Heterosigma aquashiwa is a relatively fast swimming organism, on the order of 5 to 10 meters per day. The water column is about 10 to 20 meters. That might give it a fighting chance to be able to depending on how strong the mixing is within the water column. Next. Nitrogen concentrations are cut off there, but different nitrogen concentrations higher in the south, decreasing to the north. How much did that drive the event to the south? Next. Grazers and viruses, and then I think there's one more. And of course, climate forcings are things that could be influencing all of this. So we're going stepwise through exploring all these potential contributing factors using the data that we've collected as well as numerical models and meteorological data. Next. So I'm going to first just talk a little bit here about the depth and the transport piece. Next, please. So this is a relatively simple model that we set up. We have the hydrodynamics already simulated for the system, and on top of that, we put a simplified nitrogen and phytoplankton cycling model. Nitrogen was initialized as constant throughout the system, light attenuation constant throughout the system. Next, please. And asking the question, where did this bloom go to? If it was coming out of the channel near the Alameda Islands, does it have a tendency to go to the north, to the south? One of the things you can't see is we also introduced a tracer coming out of that channel, and there's plenty of tracer that goes to the north as well, but the growth tends to happen in the south. Next. And so one of our hypotheses is that average depth experienced by the organisms or the water portion strongly influences production and biomass accumulation. We're continuing to explore that. Next, please. Now going into looking at the mixing rates and as well as the heterosigma as a considering that as a swimming organism. Next. So this is a really great image that Keith took during one of the field surveys showing the heterosigma swimming around. It's a flagellated organism, and we've been looking at the mixing rates in the system, and it is mixing rates that we can extract from the model, as well as comparing those with the swimming rates. And it does appear as though that organism, that gives it enough of a competitive advantage to have its concentration higher up in the light-rich areas. We're also thinking that it's possible that its daily diel migration, depending on the timing of that relative to the floods and the ebbs of the tides, might have a potential to disproportionately transport it to some regions versus others. Next. So this is one organism among 14 or so that we're concerned about in the bay. One more, please. And like 12 of these 15 organisms are flagellates. And so we don't want to just understand heterosigma aquashiwa. We're hoping that what we can infer here is factors that actually can contribute to a bloom, behavioral aspects of certain organisms and their ability to take off and grow. And it's not that we just detect these organisms occasionally. Next, please. The data that we've been collecting over the last several years shows that these organisms are present quite frequently throughout much of the system. And so might conditions next year or in the future years set up to allow one of these to take off if heterosigma isn't quite ready to do so. Next. And okay, well, now thinking about suspended sediments and turbidity and irradiance. Next, please. So here's the chlorophyll maps that I was showing earlier. Next. And right below that now showing turbidity measured in the same locations. And if you take a look at the date for the 17th of August, there's a little pink circle drawn there. You can see in the areas where chlorophyll is at its lowest in South Bay, those are also areas where turbidity tends to be elevated. And these are substantial differences in high enough elevated turbidity that it would make substantial differences in the light penetration. And then you can see on the 25th that turbidity has decreased substantially. And that's around the time when phytoplankton biomass increased dramatically over the course of several days, further as you go further south. So a bit of evidence suggesting that turbidity and light penetration could have driven the temporal migration of the bloom and where it took off most. Next. Showing again the remote sense data for a few images here. One more. Next, please. And now showing on top of that the remote sense turbidity throughout the system. We get a similar information here with higher turbidity in the far south, gradual decline in that turbidity down to levels where the photic depth gets to a magnitude of about 3 to 4 meters, which essentially the entire shoals in South Bay is now exposed to light throughout most of the day. So the potential that turbidity and wind that may have caused that resuspension or relaxation of wind may have actually allowed for the bloom to migrate. Next, please. And next on to discussing the nitrogen concentrations. Okay, one more, please. And so now showing again chlorophyll concentrations on the top and nitrate in micromolar on the bottom. And I'll refer again to the 17th of August showing nitrate concentrations that have been drawn down substantially in the area where chlorophyll is its highest near the origin of the bloom once it made its way out of the channel. But the nitrate concentrations remaining elevated 30 to 35 micromolar south of as you go further south. And we can see again, when the biomass increased on the 25th to the south, those nitrate levels were rapidly drawn down. So nutrient concentrations weren't the thing that kept the bloom to the south. That's a transport phenomenon, and also light is a major factor. But once those conditions were relaxed enough to enable phytoplankton growth, the organisms took full advantage of it and utilized all the nitrogen in the deep subtidal areas. Next, please. And this graphic is showing, sorry again for the x-axis being cut off, but this is showing at one location where that black dot is 3 to 4 meter deep water. On the left-hand side, nitrate concentrations in micromolar starting at around 30, dropping off rapidly over the course of 1 to 2 days, around the 18th, 19th of August. And that coinciding with gradual increases in chlorophyll, rapid increases in chlorophyll. And so one of the things that I think is important about the various pieces that we brought to bear to understand the system, the mapping gives us a really great idea of what's happening system-wide, a synaptic snapshot, but we don't know necessarily what's happening between those time points. And so having the mooring data as well helps us really stitch those things together. Next. Something else that's important that we're really puzzling with here is if you think about what allows for a certain amount of production of phytoplankton biomass, you need about roughly 1 micromolar nitrate to produce 1 microgram per liter of chlorophyll. And the chlorophyll concentrations increased about roughly twice that, twice as much chlorophyll than you can readily explain by the amount of nitrogen in the system. And so we're exploring what's the potential reason for that. What we think is potentially causing that is the nitrogen sources distributed throughout the system, the wastewater treatment plants, contributing loads to the system gradually. So phytoplankton potentially taking that nitrogen up as quickly as it's being added to the water column, allowing for that growth. If you look at some of the mooring data, you can see as well, once the nitrate concentrations decrease, the chlorophyll increase rates gradually. So there's a different slope of the chlorophyll increase, suggesting that we might be entering a place where it's nitrogen limited growth rates. Next. So just a quick overview, major HAB event in August 2022. Some of the biggest impacts that we've seen in the bay in terms of impacts on water quality. The bay's high nutrient loads resulted in more severe impacts in terms of biomass, aerial extent, duration, but something else triggered the event. because those organisms have been present for the last 20 years and nutrient concentrations have been elevated for the last several decades. Potential factors that we're exploring a number of potential factors, but some of those that are leading indicators now are low suspended sediment concentrations. Many sunny days throughout the month of August and potentially calm winds that may have contributed to some of that, allowing for decreased mixing rates and an organism that can swim and take advantage of those higher light levels to survive well. That's it. Thank you.
Thanks, Dave. That was a great talk. I don't think we have time for questions, but if you have questions for Dave, send him an email. Our next presenter is Greg Gearheart from the State Water Resource Control Board Office of Information Management and Analysis. And we're pulling up Greg's slides here, but he'll be talking about the Water Reporter app and data sharing. Looks like we're ready to go now. Thanks.
Great, thank you. Hi, everyone. Thank you for having me and thank you for your attention to this important matter and all the work that's been done. I've really enjoyed hearing and learning more as I've participated. So my name is Greg Gearheart. I am the Deputy Director for Information Management and Analysis at State Water Board. It's a big mouthful. I also just call my role and our office's role as data liberators for many of the water board data ecosystems. And so I'm going to talk to you today about a project where our team developed a data ecosystem around harmful algal bloom incidents, as you've already heard about, and expanded that data ecosystem to include other partners and users and uses of that data. And we did it very deliberately and methodically. And so what you're going to hear about today is this Water Reporter app, but what the real work was many hours of talk, interviews, documentation, and then work to develop out these data models and to make them all kind of work together. So keep that in mind as we go. And I want to acknowledge that many of these slides are borrowed from my colleagues at the Water Board, Anna Holder, Marissa, and Carly, as well as the Water Reporter slides from the Commons and the Internet of Water folks. So lots of people did a lot of work and I'm just here giving the presentation. Next slide, please. This is meant to scare you kind of a slide. So this is what I inherited as my sort of assignment when I got this job about 7 years ago. I came into this office and was told to start making data accessible from the various different programs that use data systems and applications and databases. And so this is our world in a diagram. It's actually much worse than this in reality, but this was the idealized flow of how data would go into different silos and canisters within our organization. This is old, but it's not really old because we live in a legacy system world, as many of you do too. So next slide, please. Just an honor to our deity, the flying spaghetti monster, to represent the chaos. Next slide, please. So the harmful algal bloom program in 2016 grew out of the need to start addressing harmful algal blooms, freshwater and estuarine throughout California. At that time, the California cyanobacteria harmful algal bloom network had started to migrate into the Water Quality Monitoring Council environment. They'd already done a lot of their work to sort of establish common norms and develop guidance and thresholds and many of the things that you've heard about. But the State Water Board Surface Water Ambient Monitoring Program was in particularly interested in looking at the role that we could play to help coordinate incidents and risk communication around freshwater and estuarine harmful algal blooms. So we established a small P program. We borrowed from other parts of the SWAMP program, and we started developing a data ecosystem to collect incident response data. And really what that was is a very small database. I think it was even in Access at first, but we built it on the fly to sort of quickly start gathering what we needed. And in 2017, it was in use and we were starting to collect the data. In a matter of months or a year or two, we all realized that data model was not sufficient to answer all the questions and there was issues with how we were collecting it. So we immediately started working on how to make that an improved database and data ecosystem. We, along the way, also developed a strategy document that many of you are familiar with, but it was somewhere in the 2019 era that I was connected with because of a role that I play on the Internet of Water as an advisor I was connected with the opportunity for them to have a proof of value project in California that showed how building out data infrastructure was both a collaborative stakeholder process as well as a technical data science project. And so we entered into this project with them where they received funding outside of us to build the water reporter ecosystem. Next slide, please. This was driven by not only the need of the spaghetti slide that you saw earlier, but really the need from our collaborative partnerships. We had been hearing and working with partners, in particular, Restore the Delta and two of the tribes. We had a longstanding relationship of working with them to understand their harmful algal bloom data and to see how it was used or not able to be used based on data flows that were problematic. So we saw this opportunity with Water Reporter and the Internet of Water to develop a new data ecosystem that allowed these users and their uses of the data to be considered in the design of the data model and how things flowed around. So we convened this stakeholder review committee and we got their interests documented in a really excellent document. If you ever want to see what I think is a gold standard of how to document these kinds of data projects, that document is a great one, I think. Next slide, please. Lots of benefits that we saw to this effort, but we certainly understood that this was something sort of new. It was something that grew out of this open data era that we're in. The ability to sort of have different partners outside of a state entity. It's very atypical for a state entity to reach out to its partners to help design the data model and the use requirements and things like that. But we really saw this as the pilot for why to do that, because the users have vital needs, business needs as to the public and to the water board's best interest to use this data better. So by doing this process the way we did, we were able to, at the very beginning, document these different perspectives and the requirements. What were some of their barriers? What are their needs? How would they use the data beyond what we thought the data might be useful for? And then how to include all that into a document and a project that included parts that are IT development within a state agency, as well as this partner entity called the Commons, who built the Water Reporter components. So this was very groundbreaking for us, and we're still in the middle of it. I would say we're near the end of the first part, but we're about to switch phases into more operational testing and what have you. So next slide, please. As I mentioned, the tribes, the Big Valley Band of Pomo Indians was one tribe and the other was a Karuk tribe. We worked closely with them at the very beginning to get their interest and input into the design. We also worked with other stakeholders, so League to Save Lake Tahoe, Restore the Delta, and the Watershed Project. Those folks all helped inform how non-governmental organizations might be interested in using the Water Reporter interfaces. I'll talk about those in a second, but that was key design information for the Water Reporter. I should mention at this point maybe that Water Reporter and the Commons had already built many of the interfaces for nationwide use of community-based monitoring. And so what we were doing was a very specialized component to something that they already had most of the infrastructure built on. And then finally, we're now at the point where we're bringing those together, what the Water Reporter does with collecting the data and making it used on their interface and platforms. We're now trying to make sure that we can connect it well to the state of California databases, as well as the US EPA's Water Quality Exchange. Next slide, please. This is just some screenshots from the applications called the Water Reporter. So it has both a browser app and a phone app interface. So you can upload photos and things like that, but this is just an example. There's mapping functions. A lot of these data integrations are automated so that, when you upload a photograph, it basically assumes and asks you if you want to include where you're at, allowing your location to be shared so you don't have to input it, things like that. Next slide, please. And then in addition to that sort of aggregate use of the data, you can also use these two interfaces to view station-specific data. And you can look at other types of data that's maybe collected around there. So you could, in theory, have a station that not only collects harmful algal bloom or cyanobacteria related data, but also other kinds of parameters and data as well. Trash, photos of trash, chemistry, or physical measurements of the water, things like that. Next slide, please. So we put a lot of energy into this user-centered design approach, and that was intentional. That's definitely the right way to do it, in my opinion. You gain so much deeper knowledge and understanding that helps inform later questions that maybe you don't have access to the people, but you sort of understood because you did such good investment in that time. And as a result, we now have a place where we're headed of a migrating of the two data ecosystems. So the state pipeline of incident reporting data should start to be interoperable with all the data that's also going to be collected and used in the water reporter interface. Next slide, please. This is just a way to bucket the types of data that's considered. You can bucket this data many different ways, but essentially there's observational data pieces that can be put in. There's on the fly. So like as you're carrying your phone, snap a picture, file a report, so to speak, into the data system. And then there's these longer terms efforts that you can upload station-specific information that includes chemistry, other kinds of parameters as well. So these are the three basic buckets for the water reporters system. Next slide, please. I like to think about this as service areas. It's also potentially a bit of a foreshadow to the data lifecycle approach to looking at this data, but essentially the water system allows the service areas to have multi-users and engaging on the interfaces. So like whether they're putting data in or getting data out, adding metadata, sharing information and data, these multi-user kinds of services are in the left-hand category. In the center, one of the key priorities of this project that we felt was valuable was there was a strong emphasis on data management. And so having like, sort of the pieces that go into place to ensure that people don't overwrite other people's records, or that you have the right kinds of fields broken into different data elements so that you can use the data as you want later. That's all considered in this approach. And then because of their background in doing community-based water quality monitoring across the nation prior to this, there's lots of emphasis in the modernized approach to that democratized data sharing. So that includes machine readable data exports, the ability to have machine-to-machine connections or application program interfaces, APIs, and then having the ability to customize maps inside of these interfaces if you don't want to download the data and make your own map. And then also the collaborative partnership energy potential is really high because of all those things and because of their emphasis on larger picture water quality things. I think there's a lot of opportunity here. Next slide, please. So the data lifecycle approach to that exact same slide would be to talk about data gets put in various ways. There's the web report form right now that comes in through the My Water Quality website. And that is how the current data gets into the Water Board's CyanoHAB database, but we are now testing the ability to also ingest data through an API with the partner web interface at the Water Reporter platform. And so that would essentially allow those two options. We are not wed to Water Reporter down the road. This is a pilot project to demonstrate how our data system could potentially have the ability to ingest data from anyone's data platform, assuming that they match our data model or do their best to do that. And we would maybe even build transform tools that would help them transform their data if it didn't match the data model going down the road. But this is a pilot for us with sort of like a generic socket approach idea on the harmful algal bloom database, blue tuna can in the middle. From there, we are now like porting out or publishing our data to the harmful algal bloom portal map, but that data also goes to the open data platforms, and we're hoping to have other ways to get that same data. So that would potentially not just be the data that we have, but the data that other folks have put into the Water Reporter ecosystem as well. And then as we build out our next version of CEDN, we call it CEDN 2.0, but as we build it out, we're hoping to really see more and more types of data do the same sort of thing. So this is a pilot for one application to share a socket going into the FHAB database, but it really is, because of the nature of the project, it's a test for how other community-based monitoring applications could share data with our larger CEDN universe on any kind of CEDN-related data, which includes physical habitat, tissue, sediment, other kinds of data types that are included in CEDN that are not considered right now. So this is a pilot for all those things. Next slide, please. And as I mentioned, we're right now hoping that the APIs from Water Reporter to Swamp, it's going to be probably like a poll system, but however we work it out, we're hoping that our state system will be able to pull in that Water Reporter data soon. We have a deadline by the end of November, I believe, to try to get that working. And then we have also some tests to get the data to flow out of that into the open data portal. We're working on a proof of concept with a new data clearinghouse or a cloud-based warehouse tool that we think is going to be vital. And then the data from Swamp, FHAB is also going to go into the Swamp data dashboard, which is maybe something you heard about separately, but it's that lower right-hand image. We can provide URLs to these things if you want. And then finally, and most importantly, we really want to center the tribes and the most vulnerable communities in California. So that's why we chose working with folks like Restore the Delta and the 2 tribes to make sure that what we're doing here is helpful in making progress towards a better data ecosystem around harmful algal blooms to get data in and to get information out. So next slide, please. And I think that's my last. Yeah, you can just click through. There might be an image that pops up, but basically this is, oops, sorry, there's not. This is how to access a frequently asked questions or a fact sheet about the product and a demo of how to use it. And I am not a pro on Water Reporter, but I'm happy to try to take questions if there are any.
Thanks, Greg. This was a great talk. Thanks for joining us. We have time for maybe one question. There are some online. One second, we're pulling up the chat real quick for questions.
Yeah, I can read the chat questions if that's helpful.
Yeah, go for it. If you want to.
Okay, so Nick, who works for me. Hi, Nick. Great presentation. How did the state team identify, recruit, or choose which external stakeholder groups to engage with early on planning efforts? I think that is a really powerful element of the project. Well, we had established relationships with some groups, and we knew that this was a bit of a lift. And so, I think we put the word out to others if anyone wanted to add into our established kind of community of practice. But I think we ended up going with ones that we knew, or at least ones that were like close, like allies to the folks that we're already working with, because it was a bit of a heavy lift and we wanted to make sure, but we also designed it to try to get different range of input, knowing that it's hard to get complete broad range, but we intentionally chose both tribes and vulnerable community, EJ interests and things like that to try to get this. And the Water Reporter team also brought a lot of their own perspective, because they had the funding that they received nationwide, and the support that they have for their project is derived from the keepers organizations, the water keepers across the country. And so they had a lot of sort of baked in knowledge about how different groups across the country would also engage. Sue Keidel, great question. Is it coordinated with How's My Waterway? It's coordinated in the sense that, I talked to folks at US EPA. I actually just noticed that the How's My Waterway website is losing some of their community face, and I don't know what's going on. So I have a question about that. But the community monitoring data that used to be on How's My Waterway is not as easy to find anymore, but we're here to work with EPA. This data does right now currently successfully flow into WQX, which is the US EPA's exchange for getting into what used to be called STORET. It's their main sort of like data system that stores water quality data. So that part is flowing right now, but I don't know how we can better flag this. I will reach out, and if you have any ideas, please reach out to me.
Thanks, that was great. I think you might transition to our panel discussion, which I believe you're on as well.
Yeah, thanks.
So at each of our, we're going to transition to our panel discussion about monitoring gaps in data integration. And if I think there's 3 panelists here in the room, including myself, that will be at the front. And there's we have Barbara and Greg joining us remotely. And then Eva is going to be our moderator. So yeah.
If I can do this, if I can. So welcome to panel for day 2. And I'm going to start with letting everybody introduce themselves and answer our opening question. So if each of you could please tell us about your background or professional experience that will inform your contributions to the conversation today, that would be great. I think we're going to start with Barbara and Greg online. So maybe start with Barbara.
Hey, good morning, and thank you for allowing us to participate remotely. Barbara Berrigan-Priya with Restore the Delta. And this is always an interesting place for me to be because I am probably the only non-scientist, non-engineer, and non-data expert participating these last 2 days. When we built our program, we deliberately went and recruited new young staff with science qualifications because you have to know what it is you do and what you don't do individually. I'm a writer and a teacher by training. However, you know, Restore the Delta is pretty much the umbrella CBO for the Delta with an emphasis on environmental justice. So we have taken in community input on HABs for years. So later on, I have a lot to say about maybe those gaps in data versus what the community reports. So it involved all of this input from the region evolved in my effort to build out a community science program that meets standards recognized by government scientists, like at the Water Board, for accuracy and best practices in collecting data and reporting. And we provide our data directly into the water reporter system so that it can be utilized by state agencies. And the other reason for building out this program, where our greatest strength is that we have a really great way of reaching people to be able to communicate our findings. So, and that's where my strength is understanding what the science part does from the staff that I work with and how they interface with state agencies, but then figuring out how do we get that information out to the public.
Thank you, Barbara. Sorry, no, that was really loud. Greg, next.
Hi, you just heard from me. So I'm Greg again, and I don't need to go back and tell you what we do, but I will just add that it's been a pleasure to work with everyone on this team, the panel members that I see, and Keith used to work in our office. It's been a real pleasure. It's a very collaborative space. And I appreciate the push that we get from folks like Barbara and the tribes to make our data systems work better for the short-term and long-term public interests that have been historically underserved, so to speak. So that is definitely one of the key reasons why we're doing this work and leaning so heavily into it is because of the racial equity work at the water boards as well.
Thank you, Greg. I'm going to let Jenna start here since she's sitting next to me.
Thanks, Eva. My name is Jenna Rinde. I'm with the Department of Fish and Wildlife. I have about 10 years experience working in the Delta now. Most of that time being on a boat or in a truck, collecting field data, whether it was water quality, phytoplankton, zooplankton, benthic organisms. Part of that role, I did a lot of data management, made sure the data was quality assured, quality control of high quality. And I was part of a lot of different teams in the interagency ecological program to promote data management in open science. More recently, at the Department of Fish and Wildlife, I am the, I'll say I was the harmful algal bloom coordinator. Since last month, I started a new position. But I was, my role was one of those funded positions, part of AB 834. And so I worked really closely with the water boards in developing the CyanoHAB program as the DFW representative. I'll pass it to Keith.
Great. Hi, everybody. Keith Bouma-Gregson. You saw I gave a talk a little bit ago. More into my background. I did my PhD in bacteria and toxic algae, and at that time I did a lot of work in rivers and worked with regional boards. Something that hasn't come up yet is the presence of benthic cyanobacteria. So when it grows as mats, it's kind of an emerging issue with public health and water quality. And so worked in that capacity and helped develop some resources for better integrating that into our response strategies. After graduate work and research, I worked, like Greg just said, worked under Greg Gearheart, leading the freshwater harmful algal bloom program for the water boards for 2 years. So was involved in a lot of the projects that Greg just discussed. And then now I'm at the US Geological Survey in more research capacity and thinking more about the ecology of these organisms and how we can understand what's driving them and what's shaping our phytoplankton communities to then inform and respond to management and mitigation actions and how we can better manage these organisms. So has sort of been on both the research and the management side of things over the last 10 years or so.
Great. Becky Stanton, I'm a staff toxicologist with the Office of Environmental Health Hazard Assessment. My background's water quality, ecotoxicology, but thankfully OEHA offered opportunity to do a full-time HABs position, which is pretty rare in the States. So I was glad to take that on. I do cover both marine and freshwater, but for the context of this, I'll stay on the freshwater side. So I've been working with Jenna and Keith at the time, and now Marissa and Carly on the FHAB program. I was involved, one thing that hasn't come up with Interstate Technology and Regulatory Council, we had the opportunity to do a harmful cyanobacterial team and then a benthic, like Keith just mentioned, benthic cyanobacteria. So help put together guidance on that. Another thing is California Cyanobacteria Harmful Algal Bloom Network, CCM, too many acronyms, but I've been co-chair with Sarah Ryan and Dave Karen and now Jamie Smith. So welcome anyone who's interested to continue discussions. We have a January meeting, we do quarterly meetings. And then I'm also involved in HAB-related illness work. So when we get any human or animal illness that might be related to harmful algal blooms statewide, we work on that. So lots of different HAB-related hats, I guess.
Thank you, everyone. I think we're going to pull up the Menti results. Okay, yeah, I was gonna say, yeah, I'm gonna have to turn around. So I haven't seen this before, I guess so. So the Mentimeters results show that the audience that participated showed the biggest barriers to access and use of HABs monitoring data are the data is in a format that's difficult to obtain, and QA/QC information is not available, and data takes too long to be uploaded, are the top three, closely followed by data collected cannot be used for what I need it for. So I would like to hear what each of you think that we can do to overcome these barriers. And again, I'll start online. Should we let Greg go first this time?
Wow, I think that's a privilege to get to go first, but I'm not sure. I mean, it's really, really interesting feedback and lots to sort of dissect in my mind from this. I'm struck by the lack of trust being such a low, and that's amazing considering the kind of things that happen in water data worlds and the Delta and places like that we're familiar with. But that's great. I think the data quality is really challenging, and I think there's there's like the data flow parts and then the data usefulness part, which is informed by the data quality. I think we can definitely add some workflows and opportunities to increase the documentation of QA, QC, both in the planning of the data collection as well as when it's actually stored and passed on. We have some experience in our program doing that with other kinds of data sets. It's typically more useful when you have the chemistry kind of data elements that allow you to sort of bin these data sets or data records into fields that'll sort of like 1, 2, 3, 4, 5 data is great, all the way down to data quality is not documented at all. We can do those kinds of things where we can, but I think it's going to be hard for some of this incident response or incident allegation data, plus some of the other visual stuff that's, I think, just part of the historic data set. It's going to be really hard to improve the QA, QC information that gets passed along with it. But going forward, we're committed in our shop to definitely improve how we do that going forward. And on the pipelines, I think there is hope on the horizon. You know, it's not a silver bullet. There's no like magic button that Water Reporter or the API that we're talking about in my slide deck. It's not going to solve the problem right away, but I think with iterative steps where we try something, find out that this is maybe working or maybe it works worse, and how do we fix it? All those feedback loops are really important to improve the data pipelines. And so I'm hopeful, especially considering the fact that, you know, you wait 6 weeks in this world and new technology comes along that makes things sometimes a little bit easier. So I'll pass it to the next speaker then.
Barbara, do you want to go next?
Yeah, so I think that the time differential between when data is taken versus loaded up is probably the first thing that maybe has to be, from our perspective, overcome. I was struck today because Dr. Hartman went back and DWR pulled all this additional data that could feed into a historical analysis of HABs. And I was thinking about, as they do that tracking going into 2023, how could it inform and help the program if all of that was also loaded into Water Tracker or depending how, or excuse me, Water Reporter, or building out a database so that we had more access to that information in real time? Because I think part of the problem is if cumulative data takes so long to be uploaded and you're getting community reports, and then as scientists, you're saying the community observations aren't matching the data, what we can't pinpoint or see right now is where are the time lapses, because you're dealing with a dynamic environment in the Delta. I'm thinking about what we observed just in one of our testing site locations this summer at Smith Canal, and how the HAB bloom started really early and then it disappeared, and then it came back and then it disappeared. So if we aren't matching up more science-based testing done by agencies or groups like ours that are getting trained in real time with what the maybe community observation is, I think there's a chance that dynamics could be changing by the time we're getting to it. I hope I'm making that clear.
Thank you, Barbara. Yeah, I think that's clear.
Okay.
So I'm going to let our in-room panelists continue.
I'm I'm just going to kind of address some of what Barbara just talked about. As someone that has been in the field doing instant response, often it's the same people that are in the field working very long days, coming back, being asked to upload the data, make sure it's high quality, and get it ready. I've been in situations of I've worked a 10, 12-hour day and needed to make a GIS map for my manager the next morning so they could have a pretty figure the next morning.
Yeah.
Not saying that we can't overcome this, it's just that is one of our challenges is our field staff, and especially in periods of drought or other like opportunities where there's a lot of field work, they're getting pulled in multiple directions, and it can be a challenge. And I think too with that is, I was seeing on this thing that it's, what was it? It's difficult to obtain, and what's something I'm really proud of within this, specifically in the last 5 years, especially in the Delta, is we're really trying to do open science. I think a lot of the challenges is where that information is, because a lot of this data we've been trying to make publicly available. I think there's a disconnect of where you can find that information.
Yes.
And we learned that really from the last drought in the 2014-2015, and that's why we had Assembly Bill 1755, is to help overcome that challenge. So we've done, I say we as the community in the Delta have done a lot of progress of making data open accessible. It's just, it's often the same people collecting data. They're the experts. And then honestly, we have lost a lot of our historical staff. We've had individuals working since 1975, the original people. And we, I called myself an interpreter. Well, basically I would sit down and ask questions just to digest, download these historical knowledge, and try to document that on a piece of paper and then call for our metadata. And that takes time. And so just to kind of give some of that perspective, I'll give it to Keith now.
Yeah, I'm now more of a data generator, so I sort of echo what Jenna said. It takes a lot of effort to get that data, back, QA'd, formatted, sort of out to other folks. Those are not. It's a non-trivial effort to really get that pipeline efficient, and I think one of my observations is that the public health aspect of HABs. The other data delivery like pipelines integration efforts weren't operating on the same time scale that the public health questions and needs are operating on in the Delta. So there's this pretty impressive and quite remarkable like environmental monitoring program and data integration efforts, but it's like a monthly or yearly scale because you're looking at decadal or multi-year changes in the Delta, and the public health need of like days, hours of data to sort of implement that. Like for us to do that in USGS, like it would it's a lot of staff time to sort of say, okay, we like now have like a 48-hour, 24-hour turnaround time. And even for labs, there's only so many labs as far as toxins. There's fewer kind of like rapid response, public health oriented labs for generating these parameters. And so even working with a laboratory that might be later, you say, oh no, now we need it actually within 5 days. They may not be able to do that. So I think that this also just echoes the theme of this whole workshop is like those monitoring goals. And I feel like one of the biggest, like sort of two categories of maybe the biggest distance between them are like the public health versus the environmental management needs and questions that might exist. So it is not surprising me if like the data formatting being difficult to obtain, taking too long to be uploaded, because those are non-trivial challenges that require dedicated resources.
Try not to reiterate things too much. Yeah, so I think Keith mentioned one of the things for when I'm wearing the public health hat is at least being aware of getting the information, even if it's just like the microcystis visual indicator, to at least have something on the map for huge areas that have a lot of data collection. But when I look at like the HAB incident map on the water board, there would be nothing there. It's like, I know there is a ton of information out there. Let's at least have something that indicates what the situation is. So I think making that connection has been good. The other part is at some point we sort of, like if we get an illness report and we it deviates out the investigation of a particular illness, because oftentimes we're hearing about it when there's, the human or animal that is resolved one way or the other, but we're trying to inform the investigation of that, as well as what does that mean for future recreators or animals at that water body. And so it sort of splits on what is the more time critical, and then what is the data integration across these data sets. And at least knowing what data sets exist and who's responsible for that, we can at least say, okay, well, we really want to pull this piece of information for this water body in this date, and at least we know who to contact. So there is a possibility of maybe we couldn't get all the data on a timely fashion, but at least we could pull and expedite, transferring that information or expediting that lab result for a particular incident. So we do have that opportunity. That doesn't necessarily help us with advisories on every water body, on a daily basis or weekly basis. And I think that does come down to the funding and staffing and all that questions that are kind of the broader issue, but at least we've tried to solve that on a particular incident basis.
Okay, I think I see Barbara has her hand raised.
Yeah, just one point I want to add that kind of builds off of this is, I know yesterday there was a lot of talk that kind of like a data standard or quality standard was maybe the first priority, I think, for people in the agencies and in science groups. The first priority for us is public notification for public health and safety in surface waterways. We are constantly contending with trying to get a large unhoused population moved away from HAB sites. We're waiting for the data to come in on what is the impact of aerosolization in our AB 617 community to put out warnings. And so in the tracking, I think there has to be kind of this dual process that's built in where you identify there's a problem and there's maybe a quick test, kind of like the water sampling tests our teams are already doing right now, where that data can be put out in a much faster fashion to protect public safety, with the idea that it's labeled as further reporting will be ongoing, because I understand that you have to then go and get water samples, look at conditions. There's all kinds of science analysis so that we get to the understanding what is happening so that eventually you get to a point of mitigation. But there is an immediacy and concern for protecting community that can't get lost in that wait. It has to be up front and center and done first. And then you have to be able to go back and build in, I think, that back research that you all need, the true experts.
I'm going to respond, or maybe it switches a little bit, but something that's come up is the microsystems index scale has been really useful with its own caveats. I know there's a whole discussion on that. And that's something that you can report out pretty quickly. The challenge in the field is there's this cultural shift in how to collect data, and we're working on ways to streamline that. A lot of surveys are working on pen and paper. And so I've asked surveys to send me photos of their field sheets, just to try to get that information, but still then it's me trying to write that down, and there's still that QA/QC element. Is it getting transcribed correctly or not? So some of the efforts going on is trying to use tablets or other ways so you can enter your data on that and try to streamline it as much as possible so that way we can get the information out. It's just taking time. But hopefully, I think we're moving in the right direction.
Did you have something to add to, Greg? I thought you made a comment in the chat or something.
Well, first I want to just support that really important observation that we have two roles here. One is to collect data for the informing of our regulatory business and our practices that serve the public that are kind of on a little bit of a longer scale than that other high priority pressing need to communicate risk. And this is actually the nature of the beast, why we have a ever-changing harmful algal bloom data ecosystem. Is the very first thing we did was design something that we thought would do the best we could to collect data and move forward on risk communication. That might mean the data that we're using to confirm that an event is happening or a certain status of it or whatever may not be considered data by other folks. It may not be something that ever would pass muster for some useful piece of data in a research project or something like that, because we had to do that to make sure that maybe visual was enough, maybe visual plus microscopy was enough, maybe it took chemistry plus DNA to actually confirm that this was an event of a certain type. And so that was the nature of why we built the system that we built. And now what we're trying to do is adapt it to match these other needs with the incident data and to blend it with these other really important data systems and sets that are evolving and adapting. So I think it's a really important observation that we can't lose sight of, because I mean, honestly, when these things blow up in my world, it's because of a human or a dog impact that nobody wants to have happen. And so we're trying to do our best as a state agency to help inform on risk.
Thank you, Greg. Did anybody here want to add anything? So I think let's see if there is a question from the audience or online.
Okay, there was a similar question online that Jenna kind of spoke to, but will read it off to see if there's any additional feedback. For the hurdle of field staff also uploading their data after long field days, would it help if field data was collected electronically in the field, so desk work transcribing data would be reduced?
I can answer that one. Yeah, that is one way to do it. There are some challenges of collecting data on a tablet or something. Is what if you drop the tablet in the water? Luckily, our technology has gotten better where we're having cases that float. If you work on a boat, you drop everything in the water at some point. So, but you could argue that you could do that with your clipboard and your paper too. So that is one way. Another way that when I was in the field is I would have like 2 shifts. I would have the field crew, and then I would have a team at back post-calling right away, uploading the data. And so when the field team was done, they could rest, but they were still kind of there in case there were any questions that needed to be needed. But then we had 2 separate components, and that's more resources.
Okay, do we have any questions in the room? Okay.
You're gonna walk out.
I guess the question is, from your perspective, do you think that your agencies support immediate data transfer, or are they focusing on QA/QC? We have a big difference here between health reporting and research, you know, and so from your perspective, is this an issue?
So I'll just quickly chime in that the Water Reporter project demonstrated that that's actually a both and in many cases. And so, for example, Big Valley Band of Pomo Indians have had historic data collection efforts that are very QA, QC throughout their data lifecycle. You know, they have QA plans, they input the QA and QC data into the data systems. And but they also have a desire to have a place where they can put the incident reporting and incident information. And so that was the design basis upon which Water Reporter sort of started, was the idea that we needed a place that could have room for both, because that's often what the case is. We have many other examples throughout the state where folks have been either collecting or are collecting and want to keep collecting water chemistry and genomic data that's for the purpose of either regulatory process or research. And it tends to always want to live side by side, as we've seen in these last 2 days, with the other data. And so that's the real challenge, I think. But the mandates to get the data in more quickly and up and available to the public more quickly are universal. It's not limited to the FHAB data ecosystem in my mind, but the challenges of legacy data systems and all that other stuff is real. It's very real.
Yeah, the USGS has institutionally prides itself on data quality and review, and there's a number of processes that are in place. Like once we collect data, that it gets reviewed and steps have to be taken before it's approved. And in a lot of ways, that's because there's been very weighty decisions are made on the data that's collected by people in the agency. What's coming by, we have a big stream gauging network across the state, and some of those discharge measurements are like foundational for water rights decisions that and lawsuits and things reside over how many CFS are passing through a particular gauging station. And so I think both the public response and the risk notification do push the envelope of that tension between credible data that you really can stand firmly on, but also timely and relevant. And those are often in tension, because you can sort of send a spreadsheet out quickly, but you don't want to have to go back and revise that number. But you can also spend too long, poring over data to the point where now by the time you distribute it, we've moved on to the next thing. And so those are conversations we've been having in our center. Folks that work with Brian and Tamara Krause have actually been sort of pushing the envelope too within the agency of getting folks to recognize some of these legacy systems that Greg's talking about. I think, so I said there's the public health, but there's also just technology. Things are changing. The ability for us to share information with one another is so rapid and so much easier now than it was 20 or 30 years ago when things required, printing out paper or PDFs. So I'd say within our agency, the way we sort of describe it is that tension between the relevance, the credibility, and the timeliness of it all. And we can't sort of sacrifice any one of those too much. But overall, I think we're trying to work through that.
Sure. I guess as a data user, for the most part, like one of the trade-offs we make is, I know there was the question about ELISA versus LCMS. And so for our perspective, we're happy to have ELISA again. We'd like to see more of the analytical standards, more information on the relative toxicity, but since we know the standards are limited and we don't know relative toxicity for a lot of these compounds, I guess we can't quantify them. We're happy to, make informed decisions on a total microcystin nodularrin, for example, with an ELISA, and move forward with that rather than wait. There are also trade-offs. I know we often work with tiered analysis to expedite, as well as for cost savings. But I think there are some things that we continue to learn about, like with picocyanobacteria, which I know has come up a little bit, but not too much. Depending on how you filter, that may not work. Depending on what kind of genetics you do, that may not work. So I think there are certainly uncertainties that we're still dealing with, even with the way we've tried to keep things efficient and keep moving forward and inform the public. So there's certainly things we're having to deal with. And, when I get asked, is it safe? I will almost always say, well, that's not really the question I'm answering. I'm focused on one piece and whether or not that is risk. But, so sort of the communication side there too.
I had a couple points to add. First off, within a couple weeks of actually beginning our in the field testing program this year, we quickly realized we needed a tablet. We know that some of the tribes that we work with struggled where they had data that was backlogged being entered, and we set ourselves up from day one that that wasn't going to happen to us. It also meant that I had to double up how many people I sent out in the field. So it hit our budget, it hit our funding, but we thought that getting data back to the state for the investment that was made in us initially was the highest priority. But it did delay us and has delayed us being able to get water reporter data that we're reporting connected into our website because of funding and time, and we have to finish that work over the winter. And then I guess visually what I think about, because I think water reporter is the right track, but when I think visually about what we'd want, we love CalEnviro screens at Restore the Delta for historical data, because when you go and you click on your census tracts, you can pull up this piece of data, and then the data gets updated regularly. And somehow I keep thinking with HABs, you almost need a hybrid of that. You need one color that deals with immediate unverified reports. And then you need to be able to click on those same spots and be able to get to what's your nitrate load? What are your water temperatures the last year? What are your ambient air conditions, temperatures? I think that the more historical data you can load into a site location with this kind of urgent data that's color-coded different for decision-making, I just think that would be extremely useful as a one-stop shop.
Thank you, Barbara. So I think we are going to move on to the next question. So we've talked about using the data and having access to it, but what do you see as the data gaps in being able to effectively make decisions regarding HABs? And maybe I won't start with Greg this time. I'll start over here.
Something that I'm not sure it's a data gap, again, it's this timely process. The satellite imagery has just been such a critical tool. And that's something I've learned a lot this last year and a half. And it's really having those networks of like, it is available, but I for the freshwater side, the FHAB satellite imagery map is really helpful to use to kind of see where blooms are and whatnot. So that is a really great tool, and I'm glad that's there. And I'm just guess looking at like Dave Senn's talk, the satellite imagery for that was very useful, but there was kind of a delay for processing that information. In terms of like data gaps, though, we've talked a lot about drivers, and then Keith showed his thing, but we really care about toxins, and that's not what we're monitoring routinely. There's not a mandated monitoring program. But what's been interesting when you look at the different data types that we're seeing is, we've talked about water grab samples, we've talked about spat, at the T-bag absorbent, we've talked about how even being at the top of the column, at the very surface of the column versus being 1 meter down is different. The other thing that we haven't really talked about, there's been research, is what's being accumulated by our food web. That's what I think Meredith talked about it yesterday a little bit, is like muscle tissue. You can determine if there's toxins present in muscles or other tissues. And that is a data gap we don't fully understand, but it's challenging because it's kind of hard to monitor and get tissue samples, unlike deploying an instrument where you can get it in real time every 15 minutes or have it pass through a cool robot to take a picture. So in terms for like data gaps, I would just say it's unclear about toxins in the food web, but then also how are these harmful cyanobacteria or algae competing with the good stuff, like the good food web. We've learned that some have gas vesicles to migrate in the column. Some are flagellates that have a tail that can swim as well. So and then they're not picky about their nitrogen source too, some of them. So it's really complicated. They have a lot of evolutionary advantages that we should know more about, I think.
Yeah, so I'm coming at this a little more, but that's what nowadays we're more approached to help answer questions, not just to make decisions. And a lot of those more management and mitigation questions of like, well, what do you think will happen if we do this, or why is this happening? So I'm coming at this more from that larger term understanding of why these blooms and these cyanobacteria are growing. And there are plenty of data gaps there, but I think a few key ones. One is none of our monitoring programs are really designed for HABs. So we still have a lot of questions on what is the best way to monitor HABs. Ted Flynn and Jenna just talked about distribution of the water column. Peggy Lehman did a lot of foundational work showing that, and like, net tows versus pump samples. Frequency, we did get some funding from Delta Science Program and the regional monitoring program before that to be adding toxin collections onto our monitoring sites. And that included more frequent collection in the summer to try and capture these more dynamic events. But what are, does the monthly monitoring that EMP does, is that the right frequency to be doing this to actually get the data that's needed for Rosie to write the reports and for us to understand how the management actions like the barrier are impacting HABs. Toxins, again, big data gap. So building out that is in that right scale. We have a lot of the high frequency stations, and you can see with the tidal movement, we've talked about the movement of things. That's really important to get that scale right. So I think in situ sensors are an important component there. And then, as Jenna said, the remote sensing data is proving to be really powerful at getting our spatial scale more correct. Because the tides in the Delta really are different than a lot of other systems. Even other coastal estuaries don't quite have the same tidal action that we have here on the West Coast. And so that the people aren't thinking in those terms or having to capture data in the same way that we do. And then building off that, this is work that Ellen Priest has got some funding to do, and Timon is the movement and sources. I think if we could get a better sense of where blooms are originating and how they're being transported around the Delta, and that could be really powerful. And this sort of builds off of Dave Senn's talk earlier, where he said, with the combination of the hydrodynamic models, some understanding of the biogeochemistry, you can do the modeling experiments of putting your tracers in and seeing, where's the bloom going? How is that? What factors are driving that? And I think if we could fill in the data gaps of like the biogeochemistry and the ecology using DNA methods to sort of track different strains and populations, the remote sensing to have the greater spatial coverage, and then getting the models right, we would have a much better, we could just do a lot more hypothesis testing and do more like forecasting and predictive modeling that would advance our knowledge a lot about how management actions might be changing the frequency or the intensity of HABs. So those are a couple of data gaps related to understanding why these things are happening.
Thanks. So I guess to kind of build and expand, one of the things that I think gets complicated is we're really talking about biology, which I love, but it's not strictly chemistry. So most of the toxins that we're talking about are intracellular. So really understanding what's allowing a particular cyanobacteria to outcompete all the other ones, or the other phytoplankton or the other organisms that might take up that water column space or available light or nutrients. And so the amount of dissolved toxin is usually not very stable in a lot of situations. So it's really critical to understand where the biology is moving around. And so dealing with, like was talking about, of not just when the bloom's at the surface, but what's happening. A lot of the ones will actually hang out at the metalimnion. They'll actually be subsurface, and then you have a water body destratify, and all of a sudden it's at the surface and a risk. And if you didn't sample below the surface, you would never see it. And sort of the difference we were talking about earlier about the 1 meter below the water sample and the surface, they both tell us something important, but it's different. And we have to understand why it's different and how that affects things. So I think doing that, again, talking about different types of samples and how you integrate all the different methods. Again, I think it's not, there's so many complications that I really think we need to talk about, like Keith mentioned, all the different methods that we can use and how there's trade-offs, but how they give us different pieces of information and how we synthesize that, because it's not one size fits all, it's different uses and how we build that together to understand the picture better. There's certainly a lot of cyanotoxins we're not measuring. We don't even have standards for, the methods are still being evolved. We're kind of, I think, where we were with mercury and PCBs probably 30 years ago. So I think trying to balance maybe some patience with knowing that we don't have all that underlying research, or we're continuing to grow that as the methodology improves. So there's certain things that we might say, look, maybe it's not worth investing, you know, tons of money yet, as we kind of make sure we're doing the right thing. So there's some trade-offs there. And I look forward to getting them remote sensing. We have a lot of narrow water bodies. We have a lot of reservoirs, channelization. And so getting some of those other satellites or other remote sensing tools, or the drones or the AUVs or the little sailboat, that was really cool, out there to be able to look at all those different areas, I think will be really critical, because as we talked about, we can't have people out all the time in all these areas. So.
Thank you. Barbara, did you want to go next?
Sure. So one thing I think that would be really great for us to develop is how to measure where we're tracking the incidence, flow and circulation conditions in real time. Because right now we're collecting water samples, we're taking temperature and salinity data, we can record air temperature, sunlight. But I think the two things we need to be able to kind of build into that immediate tracking is pollution load. Even though I know the state is doing regular tracking, but because there's a lag time for matching that up, I think it would be great if we expanded what kind of water samples we were taking to maybe expedite just an understanding of conditions, and then really figuring out how to analyze the kind of the water circulation conditions so that we can really begin to figure out when stagnant water plays a part data-wise. I think the off-season sediment tracking is really important for us to prepare for when HABs will surface later in the year. And then last, we support this kind of remote monitoring network that's permanent. It seems to me that some of the work that we're doing right now can be replaced with permanent monitors that could actually report right into Water Monitor, where we know we have recurring problems in Stockton and in Discovery Bay, because that would expedite reporting out to the community until we get to the point that we're actually doing good active mitigation. You know, and it would then allow groups like ours to expend our capacity to get in off the beaten path into other places where there may be a need for investigation.
Thank you, Barbara. Greg?
Yeah, so going last gives me the benefit of plus wanting everything everyone said, because I totally agree with all those and they're great. And I get to be a little more wild and crazy at the end here and say, I think we fall all over ourselves trying to communicate risk with even the best data collected events. And the Pacific Crest Trail story this year is a great example of that, where you have a community that desperately needs water at different places, and you have to communicate to them how to visually identify and understand risk, knowing that they can't treat it and they can't boil it, they can't filter it. And so this is this to me, it's the classic example that we are challenged with knowing how to better communicate that. And I think that's a data gap. And part of that data gap, I think, is another sort of provocative idea, which is it intersects with the work that we do at the water boards. And my passion for 30 years now at the water boards is to enhance how people enjoy swimmable waters in California. And that doesn't always mean water contact. So we have this beneficial use that's highly misunderstood, Rec 2, the non-water contact recreation beneficial use. We don't know how many people use it. We know that intersects deeply with this topic, though, with regards to people taking their dogs out to throw a stick and then having concerns about whether it's safe to actually go enjoy that non-contact water recreation. So those are my two ideas. I know that's not the only beneficial use that is useful in moving forward as a regulatory agency, but it's easily the least understood in my career. There's no real data or monitoring collection around who uses these waters. And I suspect, as we all do probably in the room, it's the #1 beneficial use in California. So I'll just leave it with that.
Thank you, Greg. Do we have any other questions online or? Yes.
All right, I'm going to read a question from online. There's a question that asks, is there data on which livestock farms rely on surface water, which could have cyanotoxins? What is the impact for livestock? We are aware that this can be a risk to livestock that might be have direct water contact or consume that. We have had fairly few livestock cases that we've even got reported in. I think that's more connection with those communities. I know my colleague Shannon Murphy has done a lot of work to try to connect in with that. Certainly we've got a link. We have a dogs and livestock page on the HABs portal, and in the livestock section, there was a pretty significant cattle mortality event in Oregon. There's a paper that's linked there. And certainly there have been other cases in the US and in the globe on that, but we don't really have a mechanism. It's also a question of prioritization. Oftentimes that might be on not publicly accessible land, not publicly recreated land. And so it's not necessarily a priority for us to support sampling. So that's also kind of a trade-off is, you know, if somebody that has livestock, potentially on a federal land lease or on the private property, you know, their point is, well, what are we getting out of reporting this into you? And we can't necessarily support sampling all those water bodies. So that is part of the tension too, I think, is you know, what is the benefit we're providing beyond the outreach as well. So.
Do we have any more questions in the room?
Okay, this is Tricia. There's a question online, but I'm off screen. That's kind of for me. So there's a question that asks, many hydrodynamic and biogeochemical variables that control HABs are relevant to the entire Delta ecosystem, as are impacts of climate change on abiotic drivers. The Delta Stewardship Council is convening multiple collaborative science teams. Can't the HABs modeling be integrated with these efforts? And I think this is a really excellent point. And it really gives us a preview for later today. I'm going to be providing an exit survey, and there's going to be an opportunity for folks to provide their contact information and their area of interest that they might be interested in participating in ongoing work as a result of this workshop. And then I think many other questions have been answered in the chat.
This is another gap that I was thinking of that sort of has come up. And I think another issue in the Delta too is this is more like the end user group. So this has come up like even in conversation with our group. So Barbara, you were talking about like Talent BioScreen and having all like the supplemental data, like the historical data. And my experience is seeing too, like you've got end users who are looking for more like a graphical user interface and a clickable map and like a more dynamic way to like visualize and experience data. And then you have data scientists and other folks who are looking for more of a code-based API. Like I want to just go to GitHub and like download the data and then do with it as I will. I think those groups aren't always talking to each other very well, and each one can get equally frustrated when they experience the other group and the data. Like where's my API? Why can't I? I got to actually use my mouse, and the person's like, I don't know how to code. What do you want me to do with this? So I think that could be maybe another way to be creating more bridges between those things, because we might be losing data by having those two sort of ways of approaching data sharing being maybe more siloed than they ought to be.
Yeah, actually, we did have some comments in the chat earlier during the talks that data needs to be available to all users, and these were items that people missed on the Menti that they should. They can't find data they know is collected, and it's difficult to find data in agency databases without knowing station codes and projects beforehand. So with that in mind, what do you think a successful publicly available HABs database looks like in an ideal world?
Something I want to address with when you're considering databases and our reporting structures is another challenge or barrier with integrating data sets is how you name your columns. How is it with a capital T? Is it with an underscore? Is it a string? Is it a character? All of that makes a big difference, and that's something we have a lot of great tools, whether it's online reporting forms. API is a great way to like merge data sets, but unless our columns are either the same or we have some sort of what's called like a crosswalk, where it's like, okay, this is something I think we've done in IEP is something we've been trying to do is, okay, this agency, they're required to have this parameter area like spelled this way, this problem, or this way, they have a code for it. But then if you go to a different agency, it's spelled differently, it's a different code. So having some sort of it's called a crosswalk. I'm trying to figure out like a line or a link to streamline all of that. So that's really helpful. But then some of the challenges are when we're using various platforms, because we really rely a lot on the public to give us information as well. And a lot of times that data we're receiving from the public, that data coming in is like a notes section. And so you'll see that people will list like, oh, I saw this many of this species, I saw this thing, and then just like a long comment. And yes, you could do like searches to like break up by commas, but these people are on their phones and they may misspell items, or it's hard to like find that information. So if like we had a dream database, we would have consistency in like naming structures and formats across. It's just I was trying to think of like if you were to think of your like closet or something, it's easier when you have it organized a certain way. If you need to find a shirt, if you have it in shirts, it's easy to go. But if you have it like in a big pile, you have to go digging for it, and that takes time. So having standardization just for the organization is really helpful.
Yeah, I echo Jenna said. There's a lot of, I'm not a database scientist or manager, so I know there's a lot of folks who are much more well versed in that. Those norms and expectations and best practices that are being developed. Yeah, I think, there's a lot of data that's being collected. I know there's a lot of work on like federated databases where you don't necessarily have to migrate everything to one place, but you're more just making sure things can talk to each other. Greg has much more expertise with that. Yeah, it seems like on kind of my previous point about the graphical user interface, I feel like there's so much data that you probably aren't ever gonna, the point and click method is gonna get overwhelmed very quickly to do it all like that. But I think there's really I think there's likely key user groups and relevant questions that would be served by maybe a more accessible way to interface with the data. So if we could have data scientists being sort of prioritized to do all the backend integration and the coding to have all that in place, and then at the same time be having groups like Barbara and other like key user groups working with web developers and app developers to take that really robust backend infrastructure and interconnectivity and develop the types of tools and products they need to make decisions or to communicate risk effectively. I feel like that would be my thought, because I think that risk communication, the tool is that's a much more dynamic and changing set of needs. But if you can have just like the real robust code base in the back end, then that just gives you the flexibility to sort of run with that in a number of directions.
I guess one other thing that came to mind, and I deal with this more on the marine side, but I think it's useful here, which is, in a moving water system with biology that also moves, either the food web you're concerned about or the cyanobacteria or the water, having a way that integrates, so it's not necessarily the point, but also the body of water moving past between points, or what might be happening. And so sometimes it's actually more interesting to sort of look at the more cumulative or integrated across area and across time, rather than individual points in space or in instantaneous data, to think about what it might be for like a mussel being attached, filtering out of a moving column of water, or the fish that are moving within a certain water depth, feeding in a certain habitat based on their ecology. So I think that kind of is another dynamic that maybe hasn't entirely come up, of sort of how to think about the spatial temporal integration for that kind of aspect.
Thank you. Barbara, did you want to go next?
I would just add that I can understand why you need to have fields created so that you have standardized input from the public. And I just have confidence that if we can all be forced to enter our data a certain way when we make an online purchase, that we could kind of apply those same principles into how we get the public to enter data into a system. I think the question it all comes down to for all these programs to happen the way that I think we're all envisioning them really has to do with funding. I think the water boards and all of our partners need to be funded fully for data integration and for a really successful HABs program. And that it's the same for community groups that could go out and do this work, because you're not going to be able to do 1,100 miles of waterways without the public being there as well. One of the things I hope that comes out of these 2 days is that there is a really thoughtful effort made going forward about working together to get the funding that's really needed. We need you to be funded to succeed with our programs.
Thank you, Barbara. So Greg, what does your publicly available database look like in an ideal world?
Well, I think there's lots of good ideas that we've heard from all the speakers, and I support them all. I think I subscribe to the school of thought that it's multiple places to get data. There's definitely an analogy to be made with our public libraries, where some people can go right in and they know how to get into the back room and research on their own. Other people they need help orienting at the front desk. Some folks maybe can use the Dewey Decimal card thing or whatever. So like there's lots of different sort of metaphors within there that I think apply to the open data world that I've been working in for 7 years now. And I think we as government have a lot of work to do because we put a lot of energy and faith into this idea that if we make our data open, then people will use it. But because of the things that Jenna and other folks said, like your vocabulary, your data dictionary is completely useless. Even though you made a dictionary, we don't understand your field names. There's no crosswalk. It's hard for us to understand that. And so we have a lot of work to do because I think we invested many millions of dollars across the country in open data at different levels of government for the purpose of improving transparency of government and making ourselves more accountable. And this topic very much intersects with that larger picture in my mind. It doesn't necessarily mean though that we need to just build a map. And I think that's, I feel like there's still that desire from the majority of the users of the data out there would probably benefit from just having a good map. But that doesn't solve the actual problem of making our data in different ways available to different user groups and well documented and curated. Like sometimes it's all the data in a giant accessible API kind of a thing. Other times it's let's create curated data streams. We know people like this, and let's put it out daily or weekly or whatever is the right frequency, and really think about how we describe that data and make it useful to people, and maybe even synthesize it with other data so that they don't have to do it themselves. So these are the kind of data products that I'm trying to think of in the large scale. And I think it absolutely very much applies to the harmful algal bloom topic. And it's a priority for us. So I don't have a single answer, but I think we're getting better at the water board with developing these different sort of testing our hypotheses of this might work, and then finding out, does it work? No, let's change it. Let's add to it. Let's do a better metadata. So.
Thank you, everyone. I think we're at time, so unless somebody has something they want to add. No? Okay. Then we will end today's panel and say thank you very much to all of you who participated and all of you who listened, and we're gonna go to lunch. Returning at 1:30.
Thank you, everybody.
So Eric, please break us up into the remaining participants into the top 3 groups. And what we'll do, same thing as yesterday, there'll be a mural board, it'll be the same as it was yesterday, but with different questions, and you will have a facilitator to help walk you through the mural board and end up with your report out. So we have this activity until 2:45 and then a 15-minute break like yesterday. I feel that maybe some people felt like that was too much time, so you can feel free to return to the main Zoom room at that whenever you're finished and take a break for that time, and we'll all reconvene at 3 o'clock. And some people may not have had enough time. Same as yesterday, just to make sure everyone has access to the mural board. Facilitators, please place your link to the board that was provided to you. Please provide that link in the chat, and we'll come in and make sure that it is working for all the participants. All right, so we're good to go. So 3 rooms. Thanks, Eric.
Tricia, there's 37 participants. I just want to make sure.
I think it's okay.
Okay.
Yeah. Yesterday we ended up with some small rooms, so let's just do 3 big-ish rooms.
Got it. Give me a couple extra seconds.
Okay.
Tricia, who are the facilitators today?
Yes, so virtual facilitators are going to be Chelsea Batavia, Stephen Elser, and Sylvia Engels.
Great, thank you.
Yeah.
That you pick them all into one group. The late crowd.
Just let them spread out. They only can see. Team. Oh, that's what I said for. Oh, okay, well, I did not bring my guide today, but I can. Oh, no, that's okay. Actually, yeah, there's, where the guys, so. And you're all the way down the end. Okay. Liz, when you're ready, you're in the middle.
Hey, Eric, this is, oh gosh.
Yes, I'm here.
Sorry, my voice gets projected in the whole room, but can you make me a co-host, please?
Of course.
Thank you.
You should have it now.
Gonna close the breakout room and bring everyone back together. We have another minute before everyone is back in the main Zoom room. Yeah, 30 seconds, so we'll wait. Okay, I think everyone is back from their breakout rooms. Thank you everyone for your attention. I know we're at the end of two pretty long, dense days. I am going to turn on my video so you can see people in the room. All right. And you can take down the exit survey, but I'm going to put this slide up for our exit survey at the end of my little moderating of report outs, and then before we go to Laurel, so people will have a chance to take this survey, but can you take down the slide for right now? Okay, great. And we're just looking at ourselves in the room. People online are looking at us. Okay, so I'm going to do the same thing that I did yesterday. We have only 6 rooms, so I think it will go pretty quickly, but we have 1 online room and 1 in-person room that answer the same questions. So we'll start with question 1, and I'll start with our in-person moderator if she's here. She's over here. Are you doing the report out, Janice? Okay.
Thanks, Tricia. So our question, the overarching question was, what is the eventual goal for sharing data? For example, increased data use, increased public awareness, increased transparency, make better decisions. And our group coalesced around better decisions, whether they are for public health or or management. And then how will data be used by your group? Do you know where to find data to meet your needs? Again, decision-making, where you use the data, again, decision-making was as the top of the list. Also, identify data gaps for planning future work. I was the regulator in the group, so we would use data for water quality assessment. And frankly, all data we would recognize as the HABs data, there's not enough of it, it's all useful. Do you always know where to find data? No, and part of that is we don't have a coordinated strategy. So if you're depending on getting data from projects that are going on, you need to know what those are, and you don't always know who to ask. What are resources that participants have accessed for HABs information? There are a variety. I mean, people, not just the HABs data, but recognizing the ancillary data from hydrodynamics and environmental conditions to also modeling data. But the FHAB reports, the CCHAB network portal that popped up, people would like to be able to get data from there. So that was high also on the list of improvements. Can you get the toxin data, particularly out of there, because it's one of the only places to get it, unless you know who's doing work. What are suggestions for improvement? Gee, it'd be great to have a one-stop shop for resource sharing and knowing who's doing what. And then the last question, how can we take different monitoring interests and plug them into ongoing work? Well, gee, I hear there's a strategy coming up. That would be nice to have. But also we recognize there's a need for funding and to not just having the strategy, but having an ongoing work group that facilitates getting information out, and maybe even an annual event to share data with the public.
That was excellent. Thank you so much. I want to go to room Q1A online, who was answering the same question. It's moderated by Chelsea.
I don't think we ever identified ourselves as room Q1A. That's exciting. We have a title. Hi. Yeah, so we had a great discussion and a lot of similar themes to what we heard from the in-person breakout, but I'll just summarize question by question. So the eventual goal, I think one overarching point was that there are going to be different goals for different users. And so if we can make data widely accessible for a myriad of uses so that everybody can achieve their different goals, that would be really beneficial. In terms of some of those specific users and uses, we identified management. So, sharing data to inform management of HABs for public safety and the environment. Scientific users, so informing research needs, enhancing our scientific understanding of HABs. And then the public, raising awareness and educating the public for purposes of safety and well-being. How will data be used? We had a few major categories that we clustered to understand the drivers of HABs, to understand the impacts of HABs, and also the impacts of HABs management actions, to meet various management and regulatory needs. And then as far as where you find data to meet your needs, I think we got to some of that in the next question, but there were a couple notes that it is currently pretty challenging to get data, and it would be really useful to have all the data in one location, as we heard from the last reporter. Resources, again, in the breakout, we had a lot of specific resources that were named, like the HABs portal, EDI, CDEC, SFEI, satellite data tool, a bunch of others. And a lot of these, there were also specific suggestions for ways to improve those specific tools. So those are all cataloged on the mural. I won't go through them now for time's sake. And then there were also some suggestions for, programs or actions that are not currently in place. So for example, There was a suggestion to put together a HABs newsletter. We had some discussion about sort of systematically providing on website site-specific info for example, parks in the Delta region about HABs events, and it sounds like some of that is being happened for through East Bay Regional Parks, but making that more of a standard practice. And then there was a comment about needing more info about airborne exposure to HABs toxins, and then a few other suggestions. So those are also on the mural board. How can we take monitoring interests, different monitoring interests, and plug them into ongoing work? I think that one big recommendation was just engagement of stakeholders in management and the public and other interested parties, get them involved in designing the monitoring program and then going back to them regularly to consult about how it's working and iteratively adapt the program. Another big comment was just the need to make things easy to access or make it easy to share data, and then also easy and appealing to access data. The need for resources came up to design in the design of the program, the monitoring program, and then also needing to provide resources for users of monitoring data so that they know what they're doing and how to access information. And then one other point that came up was potential concerns about data ownership and sharing, especially for academics who might be looking to publish their data later. It'd be good to kind of preemptively have a plan for how data ownership is going to work. Quickly, two other additional points that came out at the end. We had a nice little sidebar conversation about how this conversation to be a little bit more granular about specific tools that we need, whereas the larger workshop has seemed to be more generally about designing a strategy. But then there was a counterpoint that, well, if you're designing a strategy, that implies that you want to know what tools you're going to need to achieve the strategy or to make the strategy work. So that was just kind of an interesting question about what goes into the strategy and how much detail you need. And then one other point that came out, we talked a lot about agency roles and responsibilities. And I think we had a good discussion about how sometimes there might be HABs that are, linked to a specific agency's actions. And in that case, they would be the party who's responsible for, monitoring and mitigating. But, this kind of like the bigger issue of increasing frequency of HABs in the Delta that doesn't necessarily connect or can't be tied to any specific agency. It's much more of a shared responsibility, and that's why we're having this conversation about interagency coordination. And I think that we agreed that we need to, as part of this strategy, clearly identify what the different agencies' roles and responsibilities are in that larger coordinated effort. Yeah, that was our group. If anybody else in the group wants to chime in, please do.
Thanks, Chelsea. That was super comprehensive. I'm also excited to see all the individual notes that people left in the mural boards and in person. All right. So next we'll go to question 2, and our in-person moderator was Liz.
Thanks. Our question was, what are data gaps in being able to effectively make decisions regarding HABs across 3 timescales, short-term, medium-term, and long-term, in terms of what can be done right now. We discussed that there is a push and pull in terms of data that's typically collected for research and publication versus the immediate needs for public health announcements, and that's a big obstacle. Another challenge or data gap, I suppose, as it related specifically to some of the remote sensing technologies. It seems as though there are workarounds, some of the hyperspectral tools that Keith brought up earlier today could get around this, but one challenge is the satellite resolution in some of these remote or narrower channels or backwater sloughs in the system. the remote sensing isn't able to pick up that data. We spoke to more coordination between agencies to define what their short-term, their objectives are on these different timescales. We recognize that incident reports use a lot of resources, but certainly longer-term goals are needed to identify and implement some of the objectives of these various agencies. In terms of medium-term management data gaps, we discussed that getting a handle on some of the antecedent conditions and is necessary, and this kind of goes back to some of the folks that are acquiring the data don't always have time to analyze that data. So having a bit of time to review, perhaps review those relationships in the medium term could effectively assist with management decisions. Generally, this has been covered over the last few days, that toxin data is not real time, but we do know that there have been some established relationships between some real-time parameters like algal fluorescence and pH that could, augment some of the short-term reporting and potentially be used for predictive purposes. One really interesting point that came up was something that occurs regularly at CDFW with the smelt and salmon work teams. We thought this could potentially be a model in the short term as it relates to HABs and water quality parameters, but what occurs here is that there are these weekly work groups that get together, and it's in terms of getting a handle on how water operations are affecting fish salvage and fish entrainment. That's something that's specific to some of the incident take permits, but we thought it might be an effective model for some of the HABs work that's currently that folks are interested in coming together for. And yes, just in terms of real-time reporting on some of the water operations and extreme events like reservoir releases, atmospheric rivers and droughts. To the best of our knowledge, there isn't like one central location to get that data. I did learn that the California Water Science Center has received some funding to have sort of a hydrologic climate dashboard, so that should be really useful in terms of folks finding a place to look for that kind of data that's available. It's just like siloed and across different platforms. And then in terms of long-term management, a big theme that came up was the lack of case and controls. This goes back to what Rosie was talking about earlier today, that it's hard to effectively evaluate a HAB when you don't have all the right data collected when there wasn't a HAB in place. So a need for more representativeness from both HAB and during HAB and non-HAB conditions. Certainly the resolution of data that is collected over short-term studies ideally would be like collected over longer-term studies, but we know that there are funding obstacles for that too. So lastly, and over the long term, certainly a coordination of labs and methods across agencies for data interoperability is paramount.
Thanks, Liz. I feel like we're already getting some themes falling out at these that are similar across these different questions. And so let's go to question 2 online, or Q2A, which was moderated by Stephen Elser.
Hello. Yeah, so we're also talking data gaps, and for short term, the main themes that emerged for us had to do with, first, there's the importance of collecting data that is relevant to your monitoring questions. So just being clear about what you're actually collecting, what purpose it's serving. Also, there was a lot of commentary about just the need to develop a tool about where to access relevant data repositories, sort of finding them in a central location so people can easily find them and download and access and use the data in a easy to find way. And then another important point that we had in the short term was just communicating the data to the public and have a clear plan on how to communicate information about HABs to the public, and doing that through different mechanisms, whether that's a website or a phone app or something like that. And related to that point, we talked about the importance of just having a consistent message and making sure that the message is getting out about HABs, whether that be from state agencies, city officials, public health organizations, NGOs, whatever, that they're all sort of saying the same thing. Because if one group is saying one thing, another group is saying another thing, then that leads to some confusion, and that can lead to some pretty bad outcomes. So just wanting to develop a consistent message across the board, and one possibility to help do that was developing maybe a monthly cross-agency or cross-stakeholder HABs team meeting to talk about the state of things and get on the same page with each other. So that's some of the stuff we talked about in the short term. In the medium term, I would say the main theme that we had was moving beyond just monitoring for the sake of letting the public know what's happening with HABs, and into informing models in order to make predictions about what we might see in the future. So we saw a lot of comments about monitoring and improving models. And then in the long term, the main themes were that we basically wanted to use the data that we've collected through those short and medium term strategies to actually inform policy measures and implementations and regulations. And also to review the data that has been collected to further identify gaps and make adjustments based on what gaps we're observing, and to plan special research studies to address those gaps that have been identified. And then finally, we want to actually, in that long term, be able to identify funding mechanisms to actually address the implementation issues that we want to see addressed. That was something that came up a couple of times, just how we're really resource limited when it comes to a lot of this. So identifying funding mechanisms would really go a long way. And this is a more general point that we talked about, is just the importance of having clear expectations about HABs and the strategies surrounding them, because cyanobacteria, they've been here, they've always been here, and they will continue to be here. They're even in pristine ecosystems upstream. So we need to temper our expectations and think about strategies along a timeline. So strategies need to have these short, medium, and long-term phases, and there should be expectations surrounding each of those different phases. What we can accomplish in the short term is very different than what we can accomplish in the long term. So just adjusting our expectations for what we can realistically accomplish across different time horizons, and thinking along those terms in terms of the short, medium, long-term phases of your strategies, how we build in some flexibility, how we can adaptively manage the strategies to meet the goals that we're setting. And when we see that something doesn't go right, what can we change to make it do what we want it to do? So those are the main things that we talked about, but if anyone in my group wants to add anything, please speak up.
Thanks, Stephen. Yeah, sounds like some really great information, and also a lot of parallels to what we were discussing yesterday, I feel like, with the standardization of methods and approaches and how to compare data across groups. So next, we're on to our last question, question 3, and our in-person group was moderated by Keith. Are you doing the report out? Okay.
Thanks. Yes, we were talking about notification systems for risk and from HABs and to the public, are also a little bit of like environmental health officials. And so the information that we felt that the public need would be sort of the state of the situation, and then also some action guidance. So like currently in the state, we have caution, warning, and danger levels, and then each one of those has different sort of guidance on that, of like, should you not touch the water, should you not swim, or should you not even maybe like recreate at all in that area, as well as other information to be included could be like where to go for more information, where to report illnesses or incidents, And then also having a centralized place for all this information. So interactive and customizable maps, some of those already exist. And then in terms of the methods of communication, one of the things that came up was in the Delta, there are a lot of user groups. And so there was a sense of knowing your audience, and then for each of these different groups or affinity groups or user groups in the Delta, there might be different mechanisms of outreach you might have. And so one of them was that the marinas, the yacht clubs, boating clubs, sailing clubs tend to have their own memberships and listservs. And so if you can reach out and cultivate relationships with sort of the informal leaders within those networks, that you might then, they can then distribute, they sort of already hold some leadership and authority within those groups. People might be more inclined to listen or sort of give ear to what they share. But whether you know, how that might happen, it could be through emails. There also another thing we talked about was social media, and using that that method as well. Another thing that came with SMS messaging, text messages. Regarding signs, that would be like another thing, like within the notification system, there's also some more just like signage that gets posted. So if there were to be signs being placed, it would need to be, there are issues where they're not always located and easily visible place. Another thing that came up would be including information on another theme we talked about was, you know, there's like the immediate notification system, but then maybe also there's like more of a general awareness information dissemination about HABs, just to elevate people's overall knowledge, understanding of the topic. And so examples given like in Florida, where HABs may be a little more prominent, the general public tends to have a pretty high awareness of it. Becky Stan mentioned that they're often incorporated into the weather forecast. You like listen if it's going to rain tomorrow, and then the weather person will tell you what like the state of HABs are on the coast. And so, yeah, trying to also move that all forward there. And actually, I remember that was another thing too, was could notifications be included in other like public service announcements? Like, could you do outreach to a local TV station to ask them to, after they give the weather report, to include statements about the latest HAB data that we have in the Delta. And then in terms of audience variation, in terms of like, does this need to vary across your audiences? The answer was a definite yes. And yeah, that ultimately kind of, yeah, a theme that came up over and over again was knowing your audience, and then you're going to have like pretty tailored outreach and notification. I think that's everything. So thank you.
Thank you, Keith. I like the text message idea, push notifications. I get notifications whenever the International Space Station flies overhead, just because it's fun. And like, I always just ignore it. I never go to the International Space Station, but if it was like 4 HABs, I would know where to go this weekend. All right, so same question online. This was room Q3A, moderated by Sylvia.
Hi. So yeah, we came up with pretty, well, somehow similar also ideas and sentences, but for example, well, the appropriate notification system, well, we came up also with pretty nice amount of different methods, from website to on-site notification systems or text or emails. But this variety of, there is also a variety of audience, very diverse. We also came up with a nice cluster of examples, but basically to summarize, they have different levels of knowledge, let's say, like in literacy or language or about the HABs issue itself. But it's one thing is that it's necessary to be able to reach all of them using the different methods, like kind of like in a tailored way, like Keith was mentioning. And also it's important to encourage people, public to participate also. And basically what the informational format that the public needs would be the same, like what is safe to do, what guidance, what they can swim, they cannot swim, Also, this notification or information needs to be timely and also transparent as much as possible. But this is delicate because it's we don't want to frighten people and affect the business of the local people, right? Or businesses. Also, it would be good. We also made the analogy with we mentioned the case of Florida, and one of the participants suggested that it would be good to have some time of real-time information like we have for the weather, where you can check with your preferred method of app or whatever to see what's the HAB situation. And also the information needs to be consistent among different areas, like thresholds, same criteria on what to do and what the guidance, what are the safe actions to follow according to the threshold. So depending on the area where you go, it's the same thing. What are the methods of communication available? Again, we also came up with different methods from online maps to advisory levels posted online or also posted inside. And we'd also mentioned the notice signs. And that there is also a need of outreach that to target different people and disseminate this using all these different methods available. Regarding if these varies across the audience, so yes, there are like different needs depending on the audience, but all of them we kind of summarize that they need timely information, and the action needed varies by audience. In this case, like in terms of the density of information, for example, the public just needs to know how safe it is, and agencies might need more data, large amount of data like phytoplankton species, the type of toxins, other variables. And then also we had some additional thoughts, basically that there is a need for coordination in terms of public information, data availability, and messaging, disseminating this information to people. And we had a very good discussion and came up with some perhaps fun ideas for outreach to get to these people that don't know much about the HABs or the risks. So like microcystis mascot or a microcystis mobile or water skiing microcystis, things like that. And we also one of the participants designed like a nice feedback flow, but we're running out of time, so it's on the mural for you so you have it. And that's it.
Thanks, Sylvia. Yeah, I really appreciate a lot of the thought on communication that Sylvia just mentioned, that many other people mentioned. It was kind of a secret third theme that we had tucked into a lot of our structure to hope to get some ideas about increased communication with different groups. So I really appreciate that. We're gonna put the slide with the HABs exit survey back up, and we're going to take just 5 minutes for everyone to be able to navigate to that survey before having our lead scientist come back up and close us out. All right, I'm gonna bring us back. I just wanna give a final thanks to everyone who is participating today, especially to our presenters and panelists who did a lot of work to give us a lot of information. And thank you to everyone who attended and provided answers in the breakout rooms, especially the breakout facilitators. I know you guys did a lot of extra work. I really appreciate it. With that, I'm going to bring up our lead scientist. Thanks to all of you for sticking around to the end and contributing to the exit survey. Let me bring up my slides. That's not the beginning. I have the very difficult job of summing up all the fantastic information that so many speakers and panelists contributed to. Fun to go back and reflect on my notes and on the slides. Great. And we could go ahead and go to the next one. So I just wanted to kind of, before I get started on the wrap up, I did want to make an announcement. I had a conversation with Peter Goodwin, who's the former Delta lead scientist, now president of University of Maryland Center for Environmental Science, yesterday. And I happened to mention that we were holding this HABs workshop. He wanted me to share the information with all of you that there was just a harmful algal bloom control technologies incubator center that was funded across the University of Maryland Center for Environmental Science and Mote Marine Laboratory in Florida. It has received a large grant from NOAA. And next slide. This provides some opportunities that we might consider in this community, because this center is going to be competitively funding research projects that are oriented towards exploratory treatments for HABs. And the incubator funding will focus on the laboratory and mesocosm scale. And if you go to the next slide, there's a little bit more information about this upcoming opportunity. $1 million will be awarded per year, and it's expected that individual projects would range from 50K to 200K. And have a 6 to 12 month duration. There is a request for proposals that's coming out in March of 2022 with funding awarded in, I think that's June of 2022. So you might want to just keep that on your radar, as there could be some great opportunities for this community. Okay, I'm going to get into my wrap up now with the next slide. And I just want to kind of set the stage for this reminder and say that right now there are many conversations in the Delta that are focused on monitoring. It's a little bit of a hot topic. The Independent Science Board just in March came out with their review of the monitoring enterprise in the Sacramento-San Joaquin Delta. Monitoring at large has been a topic of discussion at the Delta Plan Interagency Implementation Committee meeting. It has been a big topic in the CAMP CSAMP interagency collaborative group. And it's also been a big topic as there has been a 6-agency redesign that has evaluated the practices adopted by the interagency ecological program. I guess some of the questions that are really on the table across these different monitoring conversations are things like, are we monitoring the right things to prepare us for a novel future and the rapid change of today? Are our programs sufficiently robust to document long-term trends and provide timely information about status? What best practices should be adopted in the design and review of monitoring programs? And the wheel that you see here is a set of best practices that the Independent Science Board recommended be adopted in new monitoring programs and or existing monitoring programs. And it's interesting, when I was listening to the breakout session reports just now, I note that Stephen Elser's group was really calling for something similar, that as we think about a HABs monitoring framework, we think about how we might review that program and identify gaps within that monitoring program. And then some other general questions that are on the table are, across the monitoring enterprise, are we coordinated? Are we minimizing duplication of effort and ensuring appropriate spatial coverage and integration across monitored variables? And so, many of these general questions are quite applicable and have been prominent to the discussions that we've had in this room over the past couple of days. Here, we've specifically spent a lot of time discussing challenges in data accessibility, data management, analysis, and synthesis, which are some of the best practices that you see in the center of this wheel. We've had a lot of conversations about stakeholders' needs and the critical role of equity, corresponding to the second bullet point within those set of best practices. And then we've had a lot of conversations about the technology, which is rapidly emerging for monitoring HABs. HABs, for many reasons, has a unique role to play in this set of conversations. And let's go ahead and advance the slide. HABs is an emerging problem. HABs have been around for decades, a few decades in this estuary, but have only been regularly occurring within the past decade plus. HABs are unlike many of the other constituents that we monitor in the sense that they're not driven by strict regulations. And I'm thinking regulations like those that govern the operation of the water projects, incidental take permits, et cetera. But this regulatory landscape for HABs is starting to emerge, and we've seen recent passage of AB 834, the provisions in the Delta Regional Monitoring Program Nutrient Research Plan, and we've also heard a call that there's an opportunity to bridge the gap between public health monitoring mandates and environmental health monitoring mandates, and those are all relevant to this regulatory landscape. HABs also play a unique role in the sense that it's driven by a really complex set of interacting variables. And there's a slide from Peggy Lehman's talk yesterday that shows just two of these variables, but certainly today we've heard about roles of temperature, hydrodynamics, nutrients. It really requires an integrative understanding of how the estuary functions as a system. HABs is also unique in the sense that it has an outsized impact on vulnerable human communities, maybe more so than many of the other constituents for which we monitor, or at least on more rapid timeframes. The change in status of these HABs events can be very rapid, and this requires that our monitoring program is responsive in near real time, and that information is disseminated to the relevant communities very rapidly. So next slide. So given all of these considerations and the urgency to develop a HABs monitoring framework, what should we prioritize? And here's just a reminder of the outcome of the Mentimeter poll from yesterday. One of the things that I see when I look at this poll is that although we do have some leaders here, a lot of people felt that all of these priorities were important. This is a fairly uniform distribution. Can you just toggle the down arrow, please?
So one of the things that seems apparent in listening to the conversations over these 2 days is that we really need to, in many senses, we need an all of the above strategy. One of the things that Trish highlighted, I think it was this morning, is that there was a little bit of surprise around the fact that the lowest response here was increased public awareness of HABs, and yet today we've heard so much conversation focused around what's needed in terms of increasing public awareness of HABs, and we've heard a lot about the importance of this issue. And so to me, that just emphasizes that this is a really challenging problem because we have many priorities in establishing a monitoring framework. Next slide. So one of the things I want to highlight is just, given the fact that so many things are important, what are some of the themes and needs that stood out the most? And my methods here are, I was taking notes, and Tabitha and Trish were taking notes, and I sat down and this afternoon looked at all 3 of these sets of notes to see if I could kind of coalesce some of these ideas. And one of the things that really stands out is that the community needs a commonly agreed upon set of standard practices. And this applies to many different aspects of a monitoring framework. It applies to dissemination of information and when you activate certain sampling protocols. So there needs to be standard practices about who to contact when a bloom occurs. And then also, once we've been documenting that bloom, how do we push that information out to the public and provide information not just on the status of the bloom, but what sort of actions need to be taken? That was one of the things that just came out in the breakout reporting that we heard just now. It's clear that there's a set of standard sampling practices that need to be adopted here. We've heard many calls for inter-lab comparisons. And one of the things that came out across several talks is that this visual index for microcystis is proving to be really valuable, a valuable set of data. It's simple to collect, it's very understandable. And yet we don't really have a good sense for what the bias is across different evaluators. There's a need to develop other types of metrics like this for other organisms, other toxins. But there does need to be some sort of standardization here. We've also heard a lot about the need for data standards. Data needs to be available in a common format. The data interfaces that we have now really aren't cutting it. They need to be more accessible to a wide range of stakeholders. They need to be searchable, screenable. They need to produce information that's relevant to both scientists and to the general public. And there might be different formats of data visualization or data availability that would apply to each of those audiences. The data need to have a consistent set of organized metadata that's also accessible to this wide range of audiences. And that's one of the common themes. Another is that we really need long-term funding for this monitoring framework that applies to data collection, analysis and synthesis, and communication. So some of the specific needs that we heard across the talks and panels are that it might be desirable to think about having permanent stations or permanent sensors in places with well-documented bloom histories where there is a high impact or proximity to human communities. There was a call for more regular and consistent collection of toxin data specifically. And one of the advantages of having a long-term monitoring program funded is that this enables us to document antecedent conditions, which might not be done if monitoring is driven by particular HABs events. So another common theme and need here is that there's a need for synthesis and integration of data across different technologies and variable types. We heard a great talk from Rosie Hartman earlier today about how multiple types of data were pulled together to tell a story about the impacts of the barrier across the West Franks Tract River on, or the West Falls River on harmful algal blooms within the Franks Tract. I think it's clear that HABs is a cross-scale challenge, and integration really needs to address this range of scales. We need to be able to coarsely resolve blooms and understand coarse transport patterns. But in certain cases, in order to understand how blooms are originating, we might also need to resolve what's going on in these fine scale channels within the delta. Other things that come into play with synthesis and integration is the understanding that we have a tidal system, and understanding that transport component is critical to understanding how blooms evolve and develop. And that transport component really makes this a unique challenge for the delta. The vertical component is also critical. We heard a need to understand not just what's happening at the surface, but at lower levels, and understand how changes in stratification, formation of stratification or destratification could have an impact on the development of blooms. And then also, in my mind, what falls under this umbrella of synthesis and integration is looking across a range of technologies. We talked about how that DWR microcensus visual index is turning out to be impactful, but it's not the only thing we need here. And then another common theme and need is just the need to understand the human communities that are affected by HABs. Who is using the waters that HABs occurred within? How is their health being impacted? How do we reach them? How do we get that critical information out to them? Next slide. And so I think some of these considerations were reflected in the Mentimeter poll from today on what some of the biggest barriers are to accessing and using HABs monitoring data. I'm not going to dwell on that too much, but if you go to the next slide, one of the things that I did want to do, and this is like my final meaty slide here, is just to highlight a few of the I'd say a little more in the weeds science action areas than those key themes that emerged on the previous slide. So, to me, it's clear that there are some science action areas that really stand out here. One is a need to understand the transport component within the delta. Where are the blooms originating? How are they being transported? We heard a need from the morning panel on rapid mechanisms for quantifying circulation in a spatially explicit way, quantifying residence times. There's the challenge of the vertical dimension as well, and the need to account for that vertical dimension in a monitoring framework. Then I really liked these three areas of investment that Keith mentioned in the panel this morning, that he feels would allow for rigorous testing of a suite of key hypotheses regarding harmful algal blooms, hypotheses that are critical to get some resolution on if we are to develop forecasting systems for HABs. So one is to fill knowledge gaps, data gaps, and understanding the biogeochemistry and ecology of HABs with DNA-based molecular methods. A second is to be able to resolve spatial distribution of HABs with remote sensing over a range of scales. So this could be Sentinel-2, this could be drones, it could be the sailboat device. And then the third thing is getting the modeling right, which is critical to understanding the transport component. So I think with that, I'm going to wrap up. It's clear that moving towards development of this monitoring framework isn't just going to come out of a single 2-day effort. There needs to be an extended community investment in this effort. And this afternoon in the breakout session, we heard several calls for regular meetings that span different agencies so that we can achieve greater coordination on how we're doing the monitoring, how we're disseminating information to stakeholders to ensure some consistency there. We also heard an idea of a potentially valuable stakeholder informational annual meeting about harmful algal blooms. And then I think it's important to make the point too that focusing on localized coordination at the scale of the delta or larger at the scale of the estuary, will set us up for successful regional coordination on HABs monitoring that extends to the statewide level. I just also think we have a tremendous opportunity, given that this monitoring conversation is at the forefront in the Delta, to really stand out in the monitoring enterprise and establish kind of what the state of the art is in advancing a coordinated monitoring framework that's responsive to rapidly developing conditions. And so I think that's the challenge I would leave this community with. So last slide, I think it's just the, oh yeah, just okay. So thank you all for your participation in a really informative workshop. We at the science program have our work cut out for us in order to distill all of the information that's on the mural boards, all the notes that emerged from the breakout sessions and panels. And our immediate next step is going to be to produce a report on this workshop that outlines some key next steps in advancing a coordinated monitoring strategy, perhaps. But thank you. And thanks. Thank you to all the Delta Science Program staff online too who moderated sessions. Just as a reminder, there is a happy hour scheduled at the bank today. Starts at 4:30.